Good afternoon, and thank you for joining us today. I'm Susan Yaki, and I'm here to welcome you to the Follette School of Public Affairs annual Spring Symposium. I'm a faculty member at the Follette School, and I'm thrilled to serve as the incoming director as well. So before we get too far into the substance of today, I want to note a few housekeeping matters. First, I hope you saw coffee and cookies outside, and please, feel free to eat and enjoy throughout. It is fine to eat here in this beautiful auditorium. Immediately after the symposium today, please join us for a reception, including appetizers and a cash bar just right outside in the lobby area as well. You're going to notice today that we've got a lot of substance to cover, and we're going sort of back to back to back. Feel free to get up, move around, stretch. Our speakers will be understanding of that. Today we're taping the symposium, and you'll find it posted on the Follette School website in a week or so. If you'd like to review something or share with others, please do so. Finally, in terms of housekeeping, I want to plug a great event that we're also holding for the Follette School next week. Our Follette School of Public Affairs board member, Kurt Culver, will be giving the Fitch Lecture next week, which is a very prestigious lecture on campus. It's going to be held on April 18th at 4:30 in the WID building, the Wisconsin Institute for Discovery. And Kurt is going to share his thoughts on what he calls purposeful leadership, which I find an intriguing title. So again, 4:30 next Wednesday. But today I want to welcome you to our 2018 Follette School Spring Policy Symposium. We're quite thrilled to bring together 6 distinguished faculty minds and researchers today. We're going to be discussing work in neuroscience, genetics, and the microbiome, and we're here to share their thoughts on both emerging policy issues, as well as some of the ethical implications of their research and others. We're going to arrange today in these 3 sessions. First, you will hear from UW-Madison professors Bobby Wolfe and Katherine Magnuson on the potential for brain science to inform public policy on poverty, especially for children. Second, Follette School professor Jason Fletcher and Professor Dan Belsky from Duke University are going to be discussing the integration of genetics with health and education research. Our last session is going to be led by Follette professor Pam Hurd, as well as UW Law professor Alta Charo. They're going to be discussing genetic information as well as data from the microbiome, and specifically talking about policy research and the ethical implications that may result. So all of our presenters are going to spend a little bit of time kind of introducing each other throughout the day. And with that, it is my great esteemed pleasure to introduce my colleagues, Professor Bobby Wolfe and Katherine Magnuson. Please join me in welcoming them.
Thank you, Susan. I'd like to welcome you as well. Delighted to see you here. And to more formally introduce my co-speaker when we're going to talk about the potential of neuroscience for policy, Katherine Magnuson, who is the Associate Director at the Institute for Research on Poverty and a professor in the School of Social Work, who focuses on child development and is involved in a really exciting new project that she's going to describe to us. So with that, let me get started. I have up here my outline so you know where I am going. I'm going to introduce briefly the how we measure poverty, just so that we were on the same page, and then turn to the topic of the day to look at the role that neuroscience can play in trying to inform policy for children who are growing up in low-income families. So first, how do we measure poverty? We use something called the Federal Poverty Line that has been in place since the beginning of the War on Poverty. These are the levels. It depends on the number in a unit. And what you can see up here are the guidelines for 2018. So for a family of 3, it's about $21,000. For a family of 4, about $25,000. A parent and child, about $16,000 $1,500, and for a single individual, it's about $12,000. So when we talk about children living in poverty, we're talking about children who are living in families whose incomes are below that level. And this is just a figure to give you some sense of how many children are poor. I actually have 3 levels up here. So the kind of medium blue is the poverty line that I just mentioned. It shows over time, so you see it was higher during the Great Recession. It's coming down somewhat now. But still, if you look at these, what you can see is that something like over 18% of children are living in families with incomes below the federal poverty line. Perhaps more shocking is the lowest group. So if you think about those already low levels of income divided in 2, and those are children who are living in what we call deep poverty. And here what you can see is that about 8% of children in this country are living in families with incomes that are in deep poverty. We would expect these would have influence on how well children do, and we will see that's exactly what research has certainly shown. If we look just briefly at ages, what you can see here, I have 2 figures. On your left, it shows broad age groups, and it indicates that the younger you are, the more likely you are to be living in poverty. So children are much more likely to be living in poverty than prime age adults. And then the group that's the least likely to be living in poverty by this measure are older adults. And on your right, it shows children by age groups, and the same pattern holds. The younger you are, the more likely you are to be living in a family that has income below the poverty line. Very briefly, in terms of this state, Tim Smeedy, who's part of the faculty at the Follette School, has done extensive research. These are rates of poverty for children in this state, and I show it to you because it indicates the very different rates by race, ethnicity. So the bottom line suggests that only about 10% of children in white families are living below the poverty line, whereas more than 1/3 of children in black families are living below the poverty line, and about 1 in 4 of those of other races. So with that as background, I'm now going to move to my main focus, which is to look at what are we learning through neuroscience and how can it inform policy. I thought I'd pause a moment because this is really a new area of how did I and the person I work most closely with, Seth Pollack, got into this area. How did we combine economics with neuroscience? Not going to talk long, but there is a serendipity about it that I always think is worth sharing. When I was on a committee at the National Academy of Sciences, I was on a committee called Youth, Families, and Children, and we held a workshop on child development. 2 interesting things happened there. One, Seth was asked to come, and he talked about his work with children who were orphans and their experiences. And Ed Moss, who is one of the researchers gathering data for this NIH data set that was using brain scans to measure the brains and learn things about the brains of healthy children, was there. And somehow through this, we came up with this idea that we could really see if we could use this tool to try to better understand what poverty was doing to children. So, a very fortunate meeting. So how do we study the brain? So there's really 3 main tools that we use for this. The first one, which is actually the bottom of of the slide in front of you, is we use EEGs. So what they used to do is measure brain waves. It's essentially the same tool that if you've had an EKG, they use for your heart. It's looking at activity. It measures the rate of activity, and that gives you a picture of the responsiveness, how rapidly people respond, how rapidly a child is responding. It's a relatively inexpensive approach and gives you an information on the speed of a child's response. The other 2 use a more complicated, expensive machine, And these are magnetic MRIs. They do two things depending upon the way they're programmed. One just uses the machine to look at structure. So in essence, what it does is it's looking at the brain in terms of layers. In these layers, there's gray matter and white matter and connectors. And because the water and the fat consistency differs, they can be read on these scans. The other tool is using the MRI for functional MRI. And here, what you have is individuals, again, they're in this tube, and they are exposed to different ideas. And when they respond, the oxygen goes to the areas of response, blood flows to those, and again, we measure that in terms of the blood flow. Unfortunately, that's relatively slow. So while we get these pictures of how an individual is responding, it is not at the speed of which an EEG. But these are the primary tools that are being used to try to look at what poverty does to a child. When we do that, we use three different kinds of information. One is structural knowledge, and you'll see in a moment that we measure the area as a child grows up of key areas that we're interested in on the basis of earlier animal studies, and I'll explain that in a moment. We're interested in functional knowledge, the kind of responses that individuals have to a variety of different types of problems that are posed or visuals that are shown to a child. And lastly, we're interested in the timing of these. When are children most sensitive? What parts of the brain are sensitive when they're 3 years old? What part is sensitive when they are 10 years old? So we're looking in part to learn the periods of response and the sensitivity in those periods. So what areas do we look at? Well, we're basically trying to look at areas that are important for cognitive types of performance. And we're looking at areas that are going to continue to develop as a child grows up. So these are not things that are formed and in place when a child is born, but rather are likely to be affected by the environment. So the areas that we have been focusing on, and again, these come largely from earlier animal studies, would be the frontal lobe, which you can think of as your executive function. It's where you make decisions, it's how you organize, so it's a critical part of your brain. The next part that we look at is the temporal lobe, which is kind of near where your ear is. It's tied to memory, to some emotion, and to your ability to use language. The parietal lobe, which is back here, is an area that we think of in terms of processing information. And it also integrates sensory information. And the last area, and the area which in some sense has gotten the most attention so far, is the hippocampus, which is internal. And that's part that is important for processing spatial and contextual information. It's important for long-term memory and also for stress responses. So those are the critical areas that individuals have been focusing on as we try to develop these ideas. I've already mentioned that we use two criteria to select these areas. So when we first started on this about a decade ago, there was not a lot that was known in terms of poverty because people just had not asked the question about what role might poverty play in affecting these areas of the brain. And one of the first studies out that Seth Pollack did with a graduate student that we worked with, Jamie Hansen, was to compare the area, this is for the hippocampus volume, to two sets of children that they had been working with earlier. One is a group that Dane County had referred for early neglect, and a second was a set that had been sent to Child Protective Services in Dane County who'd been subject to physical abuse. And we all think of these as horrible experiences for a child, so it was not initially, I think, surprising to them to see that the hippocampus area was smaller for children that had those experiences. But what was surprising is that if you look here, children who were growing up in poverty look quite like those children in terms of the smaller size of the hippocampus. Other studies that inform use an animal model, and what this slide up here suggests is that the linkages between areas, in this case the hippocampus to the prefrontal cortex, those connections are stronger, there's more of them, when you use information more regularly. But if you are under chronic stress, you're likely to have fewer of these connections. And so based on this, what we might expect is that provided the same sort of information, a child exposed to chronic stress would be less able to respond than an individual who had not been exposed to chronic stress. So this is using information from animal studies where you can actually open the animal up and see the brain. We don't quite do that with humans, so that our information sources are quite different. Well, as part of this, there have been 2 studies that have looked at when do these differences begin? Is this something that children have when they are first born, an infant, or is this something that develops through the environment? And what this study that's up here suggests is that they believe, this is using EEGs, that early on we don't see differences in terms of an infant. So regardless of the parent's income, their responses look quite similar. The study that Seth and I with 2 graduate students did found the same thing. Here we were using structural MRIs, and we found that when you looked at brain early on for young infants, you don't see any differences. Now, I still want to put a caveat on all of this. We're talking about very small brains, and so are we measuring it accurately? We don't really know. We did this research with a group in North Carolina who at the time were the only ones who had a template that would even allow you to measure these critical areas. So it's something that we think other research needs to establish, but at least to date, it looks as though these experiences and these changes occur after birth. So it's not genetics, it's not something that happened before your birth, it is the environment that individuals are in. Well, let me just briefly talk about the kind of research that Seth and I have done, and then we're going to hopefully mention 2 or 3 others. The NIH collected extensive data on a set of children that were ages 4 to 18 when they began, and they had each child, or the plan was to have each child come in 3 times and be subject to a variety of different tests, and each time they were scanned, and so we have their exact age at the time, and we have repeated measures on the same children. So it's a large sample for this kind of research because it's expensive research to do. So we have these data once they were released, and then we had to read them in terms of measuring the size, and we here is not really appropriate because it does not involve me. So I can't read your brain, but I have colleagues who can. So going on then, when we first got together and discussed how we would move forward, I asked the question, is larger better? Is smaller better? And the answer I got was, we don't know. And I said, well, I'm an economist, and unless you tell me which is better, I don't know how we're going to move forward and try to pursue the question. So the first thing we really had to do was to do norming. And that's what we did. So we took all of the children in our sample, their multiple scans, used only those who were growing up in a family whose income was at 200% of the poverty line or more, and we normed them. Interestingly, while we thought we would have different shapes for girls and boys, that was not the case. There is a size difference, but it only is an absolute, it stays the same. So once we control for that, the shape stays the same. So this happens to be for total brain gray matter. I could have brought many more slides, but the point is that they're all similar. And that is that while for most children, they are very close to the norm, for children whose incomes are below, let's say 150% of the federal poverty line, they are systematically below, about 8% below. So once we had this, we did some additional estimation. Here we are looking at the differential size for children in the first row below 150% of the federal poverty line, and the second below the federal poverty line. And what you can see is these systematic differences of smaller volume, controlling for things like birth weight, race, ethnicity, mother's education, and family size. So once we had these differences, we then went on and said, can this explain some of the gap in how well children perform in school? And as part of this study, they had conducted standardized testing, and so we had 8 different outcomes that we look at. I'm just going to show you one. So in essence, what we did is say, can the different size of the gray matter of these critical areas of the brain explain the gap? And what we find is that for the ones that we have hypothesized would do so, we can explain something like, each one explains something like 16 to 20% of the gap in these standardized scores, regardless, again, of the measure, whether it's language or math or general IQ. The bottom one I show you, because that was really our test of the kind of our null, that the amygdala, which is mainly something that's tied up to emotions, should not play the same role. And that's what we find, that it's much less, when we just take the estimate, maybe up to 6%. So our conclusions are essentially that the postnatal experience of family poverty is associated with changes in brain development, that these seem to be tied to early academic performance, and we're guessing to other outcomes as well, but we haven't tested that. That the effects are primarily seen in children who are growing up in families below 150% of the poverty line. What we have found, at least, is that above 200% of the poverty line, you don't gain a lot by being in higher income families. It's not like a continuous relationship. I've already mentioned that these regional brain differences we estimate about 8% difference, and that I have not really described the sample. I don't have time, but I'll just describe them as quite healthy. There's no children who are born prematurely, whose parents have any kind of mental conditions or a set of chronic conditions either. So then the question is, will these children respond? And all the evidence we have is that these are areas that continue to develop. So when we do these kind of estimates, it may give us targets for how to focus policy. So we're learning what is sensitive to the experience of poverty. There are several other studies. This one looks at children 13 to 22. The next 3 slides that I'm just going to show you briefly are all people who are part of a group that Seth and I have been lucky to be able to pull together. Given how expensive this research is and how new this research is, we try to get in the same room people who are doing this kind of research, who go from neuroscientists to economists, other social scientists, and policymakers, to try to get us to cooperate and be able to compare results, make the data comparable enough that either, hopefully, maybe in the future we can actually use them together, but at least we can compare our findings and be able to move forward in this area. So this just shows that this divides the population of children into different income groups, and it shows these lines never cross. So as a child gets older, the higher your income, the greater in this case, again, it's the hippocampus area. This is a very different approach. This is one that simply used New York City school children, some of whom pay for their lunch and others who do not, so a very crude indicator of income, and shows here that there's more activity in those who are in higher income families. I'm going to skip the next one. We don't know what accounts for these brain changes. There are things we would all come up with here. we haven't really gotten to the stage where we can say to you, it's the stress, it's the toxic environment, it's the inconsistency in daycare. What we're finding is evidence that suggests that poverty has real physical effects and affects the physical parts of your brain and your responses. And that's gotten a lot of attention. It means more people are paying attention to poverty, and we hope that will lead to more interventions to improve the opportunities. But the next step really is to learn more about where to intervene. So when I think about this, then these are possible roles that this kind of research might play. So the first is to find the areas of the brain and the type of responses that are sensitive to the experience of poverty and to design targeted interventions. The second is to learn when children are most responsive. So again, when to target. But the last is something that I haven't mentioned and that we don't have any evidence that in fact it will ever work, but we're optimistic, and that is shadow effects. So when I say shadow effects, what we mean is suppose there is an intervention like moving to opportunity, where a set of individual families were given the opportunity to move to a low poverty area. And at first, when children went to different schools, they essentially found no effect. But 15 to 18 years later, new studies found, using IRS data, that in fact there was like a 30% increase in income for those children who were part of this intervention. That's a shadow effect. It doesn't show up right away, but it did show up later. And if we can include that in our analysis of interventions, we can certainly allocate resources to those that are most effective, not just in the short run, but in the longer run. So with that, I am going to stop and turn it over to Katherine, who's going to talk to you mainly about this brand new intervention that maybe is going into the field today, about right now.
Maybe. Thank you, and it's not only a pleasure to be here, but it's a pleasure to get to follow Bobby, because much of what she said is going to lay the groundwork for what I'm going to talk about. And I come to this work, much like Bobby, as much more of a social scientist and as certainly not a developmental neuroscientist. So when I started graduate school and well into my early research trajectory, I was primarily interested in the question of what policies and programs could do to break the intergenerational transfer of poverty and to support the learning of poor children. So as Bobby laid out, we know that poverty is a persistent problem in our country, so it bounces around and follows the economic cycles, but it's never been lower than 13 or 14%, and for much of the last 20, 25 years has been hovering in the 18 to 24%. So it affects about a quarter, a little under a quarter of our children. It's common. We also know that it affects people differentially. And we know also that that children who are raised in poverty are often likely to go on and be poor adults themselves. And so this suggests to us that there's an opportunity to think about intervening with children and families, and maybe even if we can't bring that poverty rate down a great deal, to make a difference by increasing the chances that when children have these experiences in childhood, that they can go on to be successful adults and experience economic success. So that is what motivated me, and I tell you, I'm surprised to find myself landing in a place where I'm thinking a lot more about neuroscience, and I'm going to tell you a little bit about why that is. I think one of the really interesting things that's happened is that we've simply learned a lot more about the brain. And while you can go back to the 1960s and read great pieces of research that talk about plasticity and the importance of early experience, it's really come to the forefront recently about how sensitive brain development is and other physiological development, other systems, are to early experience, even in utero, right? But particularly in the postnatal environment. So we know that birth to 3 is a period of rapid growth. There are tremendous 100 billion neurons that are present at birth, and much of what happens in the first 3 years has to do with connecting those neurons. And that's why the experiences really matter, right? So there's a common shorthand for it, which is use it or lose it, and that talks about the myelinization of neural networks, right? So kids that have enrichment, stimulation, positive experiences tend to get their brain wired in a more concrete and better way than children that don't have those experiences. And it matters because we also know that those neural networks end up forming the basis for later learning, right? It's kind of like wiring a house. You don't start until you have a foundation in place, and then you can add on the extra things. We also know that, and this comes from animal models as well as great studies of really traumatic experiences that children have had. So think of orphans in Romania who are basically in sort of warehouse-like care conditions that Chuck Nelson and Nathan Fox have studied. We know that extreme adversity is really associated with bad brain development and bad outcomes for children. What's interesting, though, is that we know a lot less, not nothing, but a lot less about trying to understand the relationship between more typical childhood adversity, particularly in developed countries, such as our own US, right? And so the links that we see between poverty that Bobby has shown you and brain development are very suggestive that the experiences are mattering. But it's hard to sort of drill down and say that it's certainly the poverty they're experiencing that's changing their family structures and their family processes, as opposed to maybe some other things. As a social scientist, I've mostly focused on the things that I think policymakers are worried about, right? So I tend to worry about poverty being associated with achievement test scores earlier in life, grade retention, literacy, IQ, high school graduation, college attendance, college completion. And I worry about these things because all of them are indicators of successful adults, productive citizens, people who are engaged in their communities, contributing to the economy, who are making a contribution, and then able to care for their own children and their own families, right? So policymakers care a lot about these types of incomes, or these types of outcomes, because they also know that they relate to income. And they worry about how SES might affect, or how poverty might affect these things. There's also very strong literature linking poverty and SES to more general health. So, and particularly diseases of inflammation. I'm looking at some of my medical friends over there to make sure I'm saying that correctly. So thinking about things like diabetes, arthritis, hypertension, which are all reflective of stress in the immune system. So these are the things that I think the public worries about, and as a consequence, policymakers worry about. Now, you can take this list and then try and crosswalk it with the types of things that my neuroscientist friends talk about, and you'll see that they're actually different, right? So there's no part of the brain that maps onto high school achievement, or even to reading test score, right? So the first conversation that neuroscience and social science have is, well, when you say visuospatial skills, when you say executive functioning, what does that mean for these other outcomes in life? And I think we've gotten that conversation going pretty far down the line, but it suggests that the types of things that policymakers care about are multi-determined by the types of cognitive skills that the neuroscientists are interested in, right? And so there's not one, it's unlikely that there's going to be one part of the brain that we're going to go down and say, okay, it's the hippocampus, right? As poverty is affecting the hippocampus, we need to focus our efforts there. It's likely a multiple parts of the brain that together are leading to these outcomes that we don't want to see, that policymakers don't want to see. And it also turns out that, as Bobby sort of mentioned, that not all of the core cognitive processes are equally associated with SES. In collaboration with some of my colleagues, we've thought quite a bit. Bobby said we don't know why poverty, or what specifically about experiences of poverty are affecting children. And I think there are some really important hypotheses that we're working on trying to figure out. So one is that SES or poverty has an effect on children's linguistic environments. We know one of the things that happens when people are under conditions of poverty and scarcity is that they have a lot of things that they have to figure out. And it sometimes also is associated with poor mental health. So think about parental depression. It takes a lot of energy to interact with young children, for those of you that have them. It can be fun, for sure, so it's not that it's always taxing, but it takes quite a bit of energy. And one of the things we know is that low-income children generally have less experience and less exposure to rich language environments. Part of it may be differences in language skills among their caregivers, but certainly part of it is probably just the amount of interaction in language speech that goes back and forth. And some of this has become quite popular in the press. So those of us that work with sort of early learning environments and early educators, there's now a buzzword of 3 turns. Has anyone heard this? Okay, we have to get it out there more. It's basically explaining to adults, so parents and caregivers, that if you're having a conversation with a child, you should each speak 3 times, right? Because it's easiest when a child says, "Mom, is dinner ready yet?" for you to simply say, "No," and the conversation ends, right? But what happens in more affluent households is that often someone won't respond with a no answer. They'll say instead, "Do you see where the clock is?" And the child will say, "Oh, the clock says 5 o'clock." And then mom will say, "Well, what time do we usually eat?" And it becomes a back and forth conversation, which is providing the stimulation in language for the child, rather than a dead-end conversation, which basically closes down and is focused only on the information. And so the notion that a rich linguistic environment, it's not just vocabulary, it's the structure, it's the frequency, the back and forth, really leads to differences in the language portion of the brain. There's work suggesting that. We haven't nailed it down, and I think the harder part of these models is getting how do we go from poverty, an economic condition, right, the lack or the not lack of money, to the parenting process and the mother-child interactions or the father-child interactions, right? So that's as much a part of the model as it is from the language input to the brain development. The other part of the model, and Bobby talked a bit about this as well, is this notion of stress. And what's interesting to me about this model is that it started out in a very specific developmental psychology sort of way by Rand Conger called the family stress model. And it really posited that economic hardship would create conflict and depression for parents. It is very hard to not be able to take care of your family, to worry about bills, to figure out how you're going to make rent, put food on the table, all of that. That it would cause problems between caregivers, so between parents of children, and that would lead to harsh parenting and withdrawn parenting. And it was to be honest, a very circumscribed model. I think what now we are realizing, and that in turn would have negative implications. So it was first done, Glenn Elder did it in the Great Recession, but Conger really focused on farm families in Iowa following floods, where there were vast decreases in family resources and a lot of stress. And he was able to link it to adolescent high school performance, so GPA, as well as anxiety and stress of the adolescents themselves. Since that early work, that pioneering work, which is still going on, work much like it, looking at families who undergo economic stress, it's really been broadened to the notion that stress happens not only inside the family when you're poor, but that it also happens outside of the family. So if you think of things like high rates of ambient noise, crowding in the household, violence in the streets in particular, if you're living in a violent neighborhood, and a range of other ways in which stress pervades the experience of poverty, then it's not only the family that's the locus of that stress. Moreover, I think there's, and this comes along with what Bobby was saying, there's a real focus on the child's or the adolescent's experience of stress, not just the parents, right? So thinking about the physiological consequences of persistent elevated stress for children and what that might mean for brain development.
So again, the pieces to put together to get this model right are complicated. You have to not only understand how poverty is affecting parenting, but how it's affecting a larger caregiving environment and how that then affects children. And I think one of the things we know fairly well from developmental psychology and neuroscience is that children don't always react to the same environment in the same way. They bring their own strengths, resources, and resiliency. So this is the type of model that I think researchers are currently grappling with. You'll notice that SES is there, but policy isn't there, or intervention isn't there. And so one of the things that I think, if you think about 4 stages, one of the interesting things is to really think about, so if this model is right, or if it's even approximately right, what then as programmatic administrators or as policymakers, where do you put your efforts? Right? And you could say, okay, I'm going to focus in on the brain and the cognitive systems, and so I'm going to think about if I know that there's a reduction in executive functioning, perhaps we should come up with a training program that can really exercise kids' executive functioning. What are the types of curriculum that I could place in a Head Start classroom that's really going to build executive functioning skills, independent of everything else that's going on in the classroom? You could really decide that you're going to focus in on caregiving, right? You could say, okay, home visiting program. I understand that parents tend to become depressed and withdrawn from their children if they're under conditions of scarcity and poverty. Let's find ways to build in mental health counseling, depression screening. Let's really reach out to these caregivers. And even if we can't fully alleviate their depression or their stress, let's help them understand how they can better parent those children in hopes of having that be a target of intervention and a way into improving families. And then the other way, and this is one where I spend some of my time on this project that I'm about to tell you about, is thinking actually about other levers, and thinking about the levers that affect family income, right? Either through transferring cash directly or by making them more able to be economically self-sufficient, so supporting their own work efforts, by providing things that their family needs, reducing the cost of rent, a range of other ways in which policies affect families' economic well-being. And so that might also be a target for policymakers to think about. And even if people are drawn towards the more micro interventions, so you want to help parents do better under conditions of stress, or children under conditions of stress and poverty, I think it's important that the costs and benefits that may be accruing to children and to children's development are at least part of the conversation when we talk about changes to programs that are going to affect families economically. I think it's an important part of the conversation. And that's part of why, with some colleagues, we've undertaken what I like to think of as my crazy news study. So with colleagues, we're actually rolling out next week. I think it's not this week, but we're this close to starting recruitment into a project that's actually going to study what happens if you're able to give families more money. So we're providing new mothers with an unconditional cash gift, and we're going to study what happens to family life as well as what happens to children's development in their brains. It's been a long journey, so we've been working on this for 5 years. There are 5 of us that are collaborating. We have 2 economists, a developmental psychologist, a neuroscientist, and myself. So we're a well-rounded team. Unless you think that's overkill, all of us have been running sort of 100 miles an hour to make this happen, because it's an incredibly hard, I'm not bragging, but it's a hard project to pull off for a number of reasons. Not the least of which is that in order to give families money, you actually have to have money to give them. So a quick shout out to our funders. NIH is paying for the research part of the project, but we have private foundations that are contributing to the payments that are going to families. And it's been a long road of fundraising to get this to happen. So what exactly are we doing? We're taking about 1,000 poor mothers, so they'll have incomes in the year prior to giving birth below the federal poverty threshold, so think of the numbers that Bobby had put up. They're going to be in 4 urban areas, so New York, New Orleans, Omaha, and then in the Twin Cities. We're going to be giving them $333 a month for 40 months. It's a little bit more than 3 years. It adds up to $4,000 a year. we have conversations where people say, well, that doesn't sound like a lot of money. For a low-income family, that's about 20% of their income. So it is actually a fairly substantial amount. Moreover, it's relevant for policy conversations. So if you think about what the maximum EITC amount is, it's just under 4,000, I believe. It's less than a year of food stamps, but it's all within the range of transfers that happen to families. And so with some foundations, they wanted us to give $10,000 a year, and we felt like that was really not a policy-relevant test of reducing income because it was unlikely that that policy would be able to do that. We're comparing these families that are getting the cash, the $4,000 a year, with a group who are getting only $20 a month or $240. We did that because we're dispersing the money by debit card, and we felt it was really important that the difference between the two groups not be their access to sort of a savings account or a debit card per se. We're loading the money monthly, and there is absolutely no restrictions on how the money can be spent. So it's a purely unconditional cash gift. And then we are studying, collecting data on these families. There'll be information collected at birth before they even know about the gift, because they have to agree to participate in research of a longitudinal nature before they get the gift, so that we aren't coercing them into research. We've been very careful about thinking through how to do that in a way that we are not convincing people to do research in order to get money. And then we are trying to collect as much information as we possibly can about what happens to these families, including information at age 3, I'm going to jump ahead, related to their brain functioning. So you remember Bobby laid out the 3 types of brain data that can be found, and we're focusing on EEG, which is the activity of the brain, as well as more standard performance measures that are not necessarily brain-based. And I'm not going to go through all of these, but you can see there's a large number of things that we're hoping to collect from the families over the time that they're in our study. We did a pilot study. Again, I won't say much about it. In part of the reason why this darn project is complicated is that it's actually hard to give money away. So we had to be sure that if we went to a hospital and we asked a new mother, do you want to participate in research? And she says yes. And then we say, well, would you like free money? That they would say yes. They did, as it turns out. It also turns out we had to make sure that they could use the card, right? So there's lots of ways, and for those of you that are like me, you've lost a credit card in your life or a debit card. We had to make sure that people would be able to contact us, or that we would be able to get in touch with people to make sure that we could get them a new card, that they could reload it, that if there was problems with the PIN resetting, all of this could actually work. And in a 1-year study in New York City, we actually found not only that we could make it work, we could recruit people, we could follow them, they could use their money. I think the average upload, and it was a lower a month. We only loaded $100 a month for the pilot, for the larger group. It was used within 6 days of getting it, right? So it was used quickly by these mothers. And when we did studies, and I don't like to oversell this because it was a small sample and a small amount of money, they talked about it being useful. And they talked about it particularly as a gap filler, right? So my food stamps ran out, and it was great to have $20 so I could go get milk, go get whatever else they needed, or not borrow it from someone else. So you have to worry about social desirability in terms of what they believe they should be saying about the income, but they seemed to appreciate it. So just a few additional things. I think one of the hardest things about this project is giving people money and then not having other public benefits taken away. So part of the reason that we're in the 4 sites that we are in, New York City, New Orleans, Omaha, and St. Paul, is that we've had great cooperation from state and local administrators of programs, and we've been able to get either legislation or administrative rulings that excludes our gift from counting towards people's public benefits, for the most part, which means that it's actually, there's a few exceptions, so don't clap yet, which means that actually it will be a test of additional income, not a test of substitution of income for cash benefits, at least for the biggest ones. There's a few, I could talk ad nauseum, there's a few that have little wrinkles, but we feel very good about the fact that it's going to be additional income. And we think that this is going to be the first study to provide definitive evidence on the extent to which young children in particular, cognitive, emotional, and brain development is affected by increased income or reduced poverty. So they're sort of flip sides of the same coin. And we think that this, again, is going to be important information that can be used in discussions, debates about the range of policies we have that transfer money to families or take money away. And again, it's in that range. We're not naive. We don't think that this information will drastically change some of the political discourse about poverty policy, but we think that to date, for many of the conversations, there simply hasn't been sufficiently rigorous information that can be used in the conversation about the effects of income on children's developing brains. And so we do think that it will be useful. And I think I'm going to jump ahead. I think one of the things that Bobby and Seth have experienced and I've experienced is a tremendous thirst in the public conversation to hear more about brain development. So these are clips. One of them I know Bobby and Seth were prominently featured in. These are clips about our study, and our study has not even launched yet. So there are more media inquiry. I've never had media inquiries about something that hasn't even started yet, but I think it's because there's a lot of interest. And as Jackshonkoff, who's been a pioneer in thinking about the intersection of neuroscience and policy, so he's a pediatrician by training and runs the Center on the Developing Child at Harvard. There's a great PBS, now a little bit old, but a great PBS frontline on the teenage brain, and he was interviewed, and it's on their website, this transcript, and they said, well, this was a question to Jack, why do you think proponents of early childhood education use brain research to raise money for their programs? He says, well, the cynical answer is that the public responds. People pay attention. But then he also says, I have 1 minute, so I'm going to read this quote and then I'll be done. If we're concerned about the impact of early experience and interventions on children's abilities, on children's competence, on intellectual development, on how intellectual development is going on, and their progression socially, then we're talking about the brain, right? And so I think there's just an increasing awareness that if you want to understand high school achievement, if you want to understand test scores, grade retention, all of this has some basis in some part of the brain, and that the siloness, the lack of conversation across areas of study is no longer warranted, and that it may even be counterproductive. We have more to learn for sure, and that there's no one science that's going to change drastically some of the political constraints or conversations, but integrating the evidence and putting it forward in a way that tells a compelling story about programs, policies, and interventions, I think is a really great way to think about that policy and science connection. Thank you.
So we have about 5 minutes left for questions.
Hello, I work with the poor and poor families and children, and what are your checks and balance that money's going to be going to the right nutrition? We spend a lot of time educating families on nutrition, proper things to buy for children, for their families. You don't have to be rich to have a good, nutritious meal plan. So what are your checks and balance that money is going to go in the right places? There's a lot of drug use. They figure it out, usually how to use food stamps to trade for drugs. I'm not hearing any of this.
So one of the things we actually know is that rates of drug use are lower among low-income people than they are among higher-income people. So the highest rates of alcohol abuse, use, and drug use are actually middle-class white college students, for example. So that said, we are giving an unconditional gift. We believe that just because people are low income doesn't mean that they don't know how to spend their money, and that one of the strengths of giving cash is that it allows them to look across the range of needs that they have and prioritize what they might need most. So for some families, for example, in Omaha, where there's not a great public transportation system, their best investment might be getting a down payment for a car even if it's a used car. In other cities, that might not be the case. They may be better served by spending their money on childcare of a higher quality, moving to a less dangerous neighborhood. And I think one of the things that we're testing, and we'll collect a lot of information that asks about a range of beneficial ways to spend money and non-beneficial ways to spend money. But what we're testing is whether what the poor need most is more resources, or whether, in fact, that's not going to be sufficient, and the more directed and supervisory type of program is potentially more beneficial.
So I just say that people are planning different types of studies, and just to briefly mention, Seth and I are in an advanced planning stage of a different kind of study where we're going to look at the effect of a family being having a housing voucher by the time a child is age 10. And here the idea is if you provide a more stable housing environment, can we see the difference in terms of how well a child growing up in poverty is doing in terms of behavior by the time they are an adolescent?
Hi, I had a quick question. Do you have a target percentage of the federal poverty line for recruitment?
Under 100%.
Okay. Thank you.
This is a somewhat different question. I noticed one of the funders for your study is Priscilla Chan and Mark Zuckerberg. And I wonder if you want to address sort of taking money from a source that has exploited the public in order to benefit the public.
I don't in this forum, but thank you for the question.
So I know there's been lots of studies in mouse models that look at the epigenetic changes associated with stress and how that affects the epigenome and the kind of the circuitry that underlies emotional regulation, stress response, cognitive abilities. Are you doing anything to look at or thinking about looking at peripheral markers for epigenetic changes in the study? That might.
We have had conversations about it and we've thought about it. We are not yet funded to do it. But yes, many people have suggested that we collect the data even if we aren't going to do anything with it immediately. So yeah. I think one of the things we struggle with is many people have many ideas about the type of data that we should be collecting, right? And they're all great. To be clear, they're all great. So, but they come at various different price points, and so we're trying to be smart about thinking about what we should absolutely get, even if we can't do anything with it yet, and what can we preserve over time and look at later. But yes, I think yes, absolutely.
Hi. I was just curious if you looked into cognitive resilience with the effects of poverty in children.
You take that one.
How are you defining cognitive
Well,
resilience?
so for example, children who do grow up in poor environments, are there children who actually do not experience these effects of a smaller brain? And what are the factors that contribute to that?
Yeah, so my colleague Kim Noble has a great quote that she likes to say, which is, I cannot, on the basis of someone's poverty, tell you what their brain will look like, right? So indeed, there's tremendous variability in cognitive functioning, in cognitive ability, and in life outcomes, right? So there are some Horatio Alger stories of children that grow up in very deprived environments and go on to succeed well beyond, the average person. So there is a tremendous amount of, if you will, resilience. I think it's, there are some things that predict it. So good parenting, rich language environment, right? You can come up with some things. And those generally are the types of things that people target when they're thinking about trying to improve children's lives. So better parenting, better schools, more interaction early in life, less stress, definitely getting violence out of schools, out of communities, things like that.
But to sort of comment on the study that we've already done, these are children that had well-educated mothers that did not have a lot of the kind of characteristics that you might think of as children who are growing up in poverty or extreme poverty, and yet we still saw these. So yes, there may be, but I think we're a long way from being able to fully understand that, and that's a consideration or a type of question we may be able to ask at some point, but we're not close to it right now.
I mean, we both know that there's, we think about it in terms of individual variability, right? For any one individual, it varies, but on average, there are important trends, right, when you look at group differences.
Thanks. Yeah, I could ask many questions, but there's a few that stand out for me. One is, have you considered comparing these issues, brain development, cognitive skills, with a country that does have a family or child allowance program? I think Canada would probably be the most obvious example where comparison might be made. Another concern I have is, do you think that giving out this money will lead to an increase in family size or in childbirth if this money is made available? Then my last question is sort of one of, so what? I mean, even if it's established that income is a main factor, how are we going to go about increasing that for most people? I mean, that's a very hard backstop. Thanks.
Well, let me answer your first, and then, and that is, we are trying to find individuals who are conducting these kind of studies in other countries. So we now have someone who has looked at how the brain reacts to differences in income in Australia. And so far, she's just been looking at inequality, but I just spent time talking with her. We hope she'll be here next October to visit and encouraging her to look. So it is, we're trying to get other researchers to look at this question and see whether in countries with more equality we see the same kind of picture or not.
I guess I'll take the more provocative question, which is, so what? Why do this? So, you know, I think it depends on whether you think that accumulated scientific knowledge plays a role in public discourse about policymaking and program running or not. And I will say, one of the most compelling reasons to do it has been the response that we get from policymakers and from the public media. There seems to be a real interest. Somehow the talking about brain development really captures people's interest and attention in a way that simply talking about bad test scores in school, although they're still interested in that, has not. And so, you know, I don't know that there will be any direct line between my research, Bobby's, Seth, or anyone else's, and better policies. But I do hope that it becomes part of the conversation and that people have a greater understanding when they're enacting policies and changing programs, what the implications might be for children and families.
I think one more question.
It's like I was done.
Both of you have been talking about the importance of rich socialization in those early years, and I'm wondering what role those devices that we're all carrying and that are brought into the dinner table and all that, what roles those devices will have, since rich families can be hooked on those computers as well as poor families along those lines. And I'm wondering if you're tracking computer use and device use, especially with children. Perhaps that can be a part of how you designate funds, whether or not, if it's a good influence, pay for those smartphone bills and all of that, and if it's a bad influence, maybe steer people away from those monies.
Yeah, so we're not directly tracking people's cell phone usage, but I think, you know, the more general point that, and you know, before cell phones and tablets, there was TV, and there's, you know, not quite the same anymore, but the idea that the TV might be on many hours of the day and that people wouldn't be talking, they'd be instead watching the TV. So, I mean, yes, the interactions matter, and no, devices are not good for promoting a rich and learning environment for children, generally speaking, although there are some decent educational apps out there.
So I would answer it a little differently, in that you have a hypothesis that it is bad. Well, you have a question that it could be bad, and I think that in some sense, the techniques that are offered through EEGs and these different kind of scans are ways that could answer that question. So I would suggest that we don't have that answer, and it may not be part of any study we plan, but I think in terms of a technique to try to answer questions of that variety, this has the potential to answer. So I think we have to stop because the next session needs to begin, but thank you all.
Thank you.
So we'll move on to the genetics session. So I'm excited to discuss with Dan emerging research and policy considerations and the integration of genetics and social and health sciences. I'm Jason Fletcher in the Follette School here. I'll introduce myself a bit when I speak after Dan. It's my pleasure to introduce Dan Belsky from Duke. I'll say as an aside that we have a friendly battle over the 3 session organizers of who's going to generate the most interest, and I've brought in a ringer. So I've brought Dan from the outside. He best, I think, represents a part of the new generation of scholars who has integrated training in health and social sciences and genetics, PhD, postdoc, and as a faculty member. He received his PhD in 2012 from UNC Chapel Hill and has been at Duke for the last 6 years. Incredibly, he's had over 75 papers published already, most in the intersection of genetics and social science, as well as forays into describing some of the policy implications of integrating these sciences. So with that short introduction, please join me in welcoming Dan to Madison.
Well, thank you for having me. I'm honored to be the least distinguished scholar on your panel today, so I hope I have something interesting to share with you. I struggled a lot with the title for this particular presentation, so please bear with me if you don't feel like it lives up to precisely what I've written up there. I'm going to begin today by acknowledging my collaborators and supporters, lest I run out of time and forget to do it at the end. I'm at Duke University, where I work with a group led by Terry Moffat and Avshalom Caspi. There are several pictures of their collaborators there, including at the University of Otago in Dunedin, New Zealand, and the Institute of Psychiatry at King's College London, along with folks at Stanford, University of Texas at Austin, UNC, UC Boulder, Princeton, and Jason Fletcher here. So this is a rough outline of what I'm going to talk about today. I'm going to give you some background about why and how we might study genetic influences on human development, how that field of research is changing as genetics moves outside of biomedicine and into the social sciences. I'll give you a little bit of data from some of our own research, and then I'll conclude with some takeaways. And I'll try and stand in front of this microphone. I have a really hard time talking and standing still at the same time. So we'll see how it goes. Okay, so step one. This is a question that I think often has an obvious answer to the audiences I speak to, but perhaps today does not, so I'll review it. Why should we study genetics in the first place? Why should an audience who comes to a public policy talk care? And I think, the answer, to me at least, is relatively straightforward. All of the outcomes we're interested in the context of public policy, human behavior, human health, social attainments, are what we call heritable. They are influenced in some way by genetic differences between people, and what we mean by that, most specifically, is that more closely related genetic relatives more closely resemble one another than do more distant genetic relatives. So identical twins are more similar than fraternal twins, siblings are more similar than cousins, cousins more so than second cousins, and so on and so forth. DNA is special. As among the many causes of health, behavior, and social attainments, in that it comes first in the human life course, and so it is not contaminated by processes of reverse causation, at least within a single lifetime. For that reason, it may provide us with some special tools for understanding processes we may seek to modify through policies or programs to enhance human health and attainment. And then finally, the idea is that these things can provide us with clues of how to best design interventions. So I'll give a little orienting information about how I'm thinking about genetics. There are different ways to approach the genome. Years ago, the standard approach was what we would call hypothesis-driven or a candidate gene approach, in which we sought to reverse engineer the biology of whatever it was that we were studying to arrive at, let's say, a protein that was important in some process. We would find the DNA sequence that encoded that protein, that's what a gene is, and we would then look for variation within that gene to try and understand behavior. That continues to be a line of inquiry and continues to be productive. However, the bulk of attention in gene discovery these days has shifted away from this hypothesis-driven approach and toward what we'll call a hypothesis-free approach, which essentially aims at leapfrogging what we know today about biology to advance our frontiers. And the general idea is to take comprehensive surveys of genetic variation and then to sift through it to identify which elements of that variation relate to the phenotype or the outcome we'd like to study. The technology for that is something called a genome-wide association study, or GWAS. The target of GWAS is something called a single nucleotide polymorphism, or a SNP. It is a single letter change in the human DNA sequence, and you can see a picture of it up here. And the GWAS is an extremely simple exercise in which we simply compare the frequencies of these particular SNPs in people who, let's say, have a disease versus do not have a disease. We make a correction to our statistical test for the fact that we're comparing hundreds of thousands, or often now millions, of these single nucleotide polymorphisms, and that's how discovery works. Perhaps as unlikely as it seems, this enterprise has been extraordinarily productive over the last decade. I'm showing you two cartoons, one illustrating the growing volume of genome-wide association study discoveries over time. And you can't read the fine print, but the point they're making is that these discoveries are being made for an increasing variety of human diseases, anthropometric traits, now including social behaviors. The other cartoon with all the many lines on it is the 23 chromosomes of the human genome annotated with discoveries that have been made assigned to their chromosomal location, and the many colors you'll see on the picture reflect the many different varieties of traits that have been studied in these genome-wide association studies. So there are lots of discoveries from this hypothesis-free sifting of the human genome. But there is also what we'll call a translational gulf between this extraordinary volume of discovery in GWAS and actual improvements in human health and well-being. And that translational gulf sort of consists, I'll say, of three components, one of which is that there are lots of these discoveries, so there's a lot of translation that needs to be done. The individual discoveries themselves have tiny effect sizes. In fact, in the field, they're often referred to as infinitesimal. That's how small let's say, risk increments associated with one of these individual polymorphisms is going to be, fractions of a percent in a population. And then, to me, the most important element is that they typically reflect what I'll call non-obvious biology. And what that means is that these genome-wide association study discoveries tend not to fall neatly within elements of the genome whose function we already know or can predict based on our knowledge of genome biology. They're essentially scattered at random across the genome, and that poses a challenge for the way we typically follow up genetic discoveries to make biological sense of them and ultimately to achieve these advances in health and well-being. So what I'm going to suggest is essentially a sort of three shifts in the way we frame and think about genetic research that aim to resolve that translational gulf, and then I'll discuss how they apply in the study of the genetics of human social attainments. The first shift, and I'll talk about each of these three in turn, is from thinking about molecules to thinking about organisms. The second is from thinking about genotypes to thinking about genomes. And the third is from thinking about phenotypes to thinking about, for those of you who are not psychologists in the audience, I'm assuming this is an unfamiliar term, but I'll say it, nomological networks, and I'll explain what that means in just a few slides if you bear with me. So in the first case, thinking about organisms instead of molecules. So the conventional path to translation of genetic discovery is to try and build the chain of molecular causation from the DNA sequence on up. So we want to know how the DNA sequence affects the transcription of RNA, how that affects the production of a protein, how that production of protein ultimately affects some molecular process in the pathogenesis of a disease. And the reason we want to understand that molecular chain of causation is because we would like to intervene on that molecular chain of causation with a molecule, with a drug. Now, there is an alternative, and it's an especially important alternative if the kinds of interventions that we envision are not themselves molecules. So what we try to do in our work is to trace a path backward from the original target of discovery, from the phenotype, through human development to understand when and how a genetic risk discovered in a genome-wide association study becomes manifest. And the idea is that in understanding that process, we can derive some clues about how we might seek to manipulate the phenotype through a non-drug intervention. The second shift I'm suggesting is from thinking about genes or genotypes to thinking about genomes. So as we said, individual genetic discoveries made in GWAS have tiny effects. What turns out to be interesting about them, though, is that as predicted about 100 years ago, they tend to combine in a roughly additive fashion, meaning you can sum up all these tiny effects from across the genome, and in so doing, you arrive at essentially a normal distribution of genetic liability to a trait or behavior. And that's some very simple math having to do with the summing of uncorrelated quantities, but it gives us, as social and behavioral scientists, something we're much more familiar with working with, which is a normal distribution of exposure. With the advantage that that normal distribution quantifies a much larger effect size because it's the additive effects of all the many variants across the genome influencing a phenotype. I'll spend another minute on this just to point out that the technology we use to create this continuum is something called polygenic scoring, and the cartoon here is meant to illustrate how that goes, how that comes about. So there is a genome-wide association study in which you have hundreds of thousands or perhaps 1 million people. You've essentially run a number of univariate analyses where you look at frequencies of the SNP, let's say in people with the disease, people without the disease. And from that analysis, you identify the disease-associated variant and the magnitude of its association with the disease for all the SNPs in the genome. You then use that set of results as a crude scoring algorithm to assign values to the genomes of participants in a separate research study. For each of the variants, you have 0, 1, or 2 copies of the variant associated with the disease. You get one from mom and one from dad. And then you multiply that count by the effect size estimated in the GWAS, by the magnitude of association between the genotype and the phenotype. You then take the average across the few 100,000 or few million SNPs you've measured in your participants. That becomes the quantity of interest for our research. That's what takes on the normal distribution. That's what we'll use to quantify genetic differences between people. There are a number of caveats in that process having to do with how we account for differences in population background that I'll get to in a minute, but roughly that's polygenic scoring. The final shift being from thinking about discrete phenotypes to what I'll call nomological networks. And this is the idea that when we do a genome-wide association study, we picked one thing, and we're going to call that one thing our target, but it's not necessarily a discrete biological construct. It has a history in development, so across developmental time, it evolves, and it exists at what we'll call multiple levels of analysis. And those are the two axes drawn on the figure, the vertical axes describing the levels of analysis, whether you're thinking about, let's say, inside the brain, human behavior, family ecology, neighborhood context, and the x-axis, so to speak, the horizontal axis, describing life course time, let's say from childhood through late life. And so anything we're studying in GWAS can be located on this kind of a graph and will be surrounded by many other related quantities that are either causally antecedent to it, that are dependent on it, or that are simply correlated with it. And understanding how genetics relate to the elements within this nomological network can help us refine our understanding of how the genetics discovered in a genome-wide association study give rise to variation in our target of interest. Okay. So a final point of orientation before we get into the meat of things, I think of the field of genetics as principally being concerned with the question of how DNA assembles life. And we have a slightly more modest objective in what I'll call sociogenomics, which is how DNA contributes to the assembly of human life courses. The research process that we pursue follows roughly this course. There is a search or discovery exercise, a genome-wide association study, to identify which particular variants we should be interested in. We're then interested in conducting follow-up analyses that investigate the nomological network in which these genetic associations are embedded. So how do they manifest across the course of development? And then finally, the idea is if we can generate that understanding, it will be able to inform the design of specific studies to test interactions between genomes and environments, the results of which can inform programs and policies that aim to improve human health, well-being, and advance social goods. Now, I want to clarify that I'm not interested in genetically informed policy development, because I think genotypes can guide us to individuals who should be the targets of our interventions. Rather, I think genetic analysis can inform us about processes which should themselves be the targets of analysis. And those processes being targeted should have benefit essentially for everyone, regardless of their particular genotype. I'll return to this at the end. Now, most of the work that we do is focused in this middle stage. I'm not a genetic epidemiologist who conducts genome-wide association studies. I try to follow up the results from those studies to understand what they mean and to try and advance this translational agenda. This work began for me with this study of classic public health problems, obesity, smoking, eventually asthma, in which we were interested in how genetic discoveries made largely in samples of adults related to processes of child development, which we know give rise to these outcomes, and the findings from those studies were broadly consistent in showing that genetic risks discovered in studies of adults are manifest early in child development, and those early developmental manifestations prove critical to subsequent risk. In other words, children who don't develop these early developmental manifestations of genetic risk largely don't show the genetic risk across the rest of their lives, suggesting potentially opportunities for intervention targeting early life processes. What I want to talk about today, though, is what happened next. So beginning in 2013, genome-wide association studies took an important turn for the social and behavioral sciences with a genome-wide association study of distinctly non-biomedical phenotype educational attainment in about 100,000 people. And the goal was not to identify proximate genetic causes of differences in education. There is no direct path between my DNA and how much schooling I complete. The idea is that by doing GWAS of educational attainment, we could learn something about genetics that influence the characteristics and behaviors that prove advantageous to people in schooling. This was followed up in 2016 with a replication in 300,000 people, expanding the scope of discovery. And there's a GWAS of 1 million people that we should all be seeing in, I would guess, the next month or so. Dan Benjamin, David Cesarini, and Philip Kohlinger were sort of the originators of this consortium, and their sort of next generation is also pictured up there: Isu Akbe, Patrick Turley, and James Lee. So I want to acknowledge their contribution to this field. They are, in some sense, its founders. I also want to acknowledge that when we talk about genetics and educational attainment, we make people uncomfortable. I thought this article by David Reich in the New York Times recently, which was about a somewhat different question, had a very useful and incisive quote about the situation. He writes, "Finding genetic influences on a propensity for disease is one thing, but looking for such influences on behavior and cognition is another. There is a way in which we feel like it's okay for there to be genetic differences between people in the risk for cancer or heart disease, but we're much less comfortable with there being genetic differences between people if they affect behaviors or characteristics that we think are essential to social success." And the reason is that we have this ugly history of the intersection of genetics and the social sciences that I think burdens this discourse and, in fact, our research agenda. 100 years ago, when geneticists started thinking about social problems, we got the eugenics movement, in which important discoveries in genetics were essentially used as cover for a racist enterprise, the worst manifestation of which was genocide. So that's a heavy weight to carry. I think that, that said, we need not abandon our research project because we will inevitably fall down that path. In fact, and I think there's a nice argument that was made by Eric Turkheimer, Paige Harden, and Richard Nisbet in Vox recently in response to another provocation by Charles Murray, in which they sort of outline why being concerned about racism as a consequence of genetic research is perhaps somewhat unfounded. And so at issue here is that these human characteristics and behaviors, things like intelligence that we know are important for success in life, are influenced by genetics. They are much more similar in genetically identical twins than in fraternal twins, in siblings than cousins, and so on and so forth. We can measure intelligence. There are genetic influences on it. There are also, as David Reich in his New York Times article pointed out, ancestry-dependent genetic differences in human populations that are correlated with the social constructs that we call race and ethnicity. And intelligence does vary, let's say in this United States population, across those race-ethnic lines. But just because those things may be true doesn't mean they have anything to do with one another. There's no reason we should suppose that because ancestry is genetic and because intelligence also is partly genetic, that those two things overlap in a way that should prevent us from pursuing this line of inquiry. And I'm happy to engage with this further in the discussion period. But I think it's important that there's a certain extent to which concern about genetic research into human individual differences in characteristics that affect socioeconomic attainments are presuming a racist hypothesis that if we look, we will find this uncomfortable truth. I think that's simply not the case. What I'm interested in fact, is something quite different. These are a couple of pieces that I've written in different places about why we're studying this, why we think it's important, but ultimately what we're interested in doing is understanding the developmental processes that connect DNA sequence with differences in life outcomes, and the idea being that those processes themselves can become targets for interventions that aim to improve attainments across the population. So for example, we published this study a couple of years ago in Psychological Science following up the first iteration of this genome-wide association study for educational attainment, in which one of the things we were interested in was how do these genetics discovered in association studies of adults, looking backward on how much schooling they had completed, relate to processes of childhood development. And what we found was that these genetic risks, as the genetic risks we observed from study GWAS of obesity, GWAS of smoking, manifest early in development. So kids who carry more education-associated alleles, more of these education-associated genetic variants, are talking and reading earlier in life. They show a more rapid pace of cognitive development. They show enhanced levels of what I've learned to call non-cognitive skills, so self-control, interpersonal skillfulness. But there was also some specificity in these results in that these genetics were not associated with children's physical health, whether we measured it when they were in adolescence or whether we measured it very early on in terms of how old they were when they learned to feed themselves, when they learned to potty train, and so forth. We've also learned that these genetics are socially patterned. That is, kids who grow up in rich families tend to carry more of these education-associated alleles than kids who grow up in poor families. And that begs the question, what does it mean to make genetic discoveries for educational attainment? So I think the presumption is, from the genetic side, that when we do these genetic association studies, we're going to identify what they call direct genetic effects. In other words, relationships between DNA sequence variants that directly influence the development of traits and behaviors that advantage individuals in their socioeconomic attainments throughout life. A second possibility is that there are indirect genetic effects in which genetics shape traits and behaviors that influence the success of a person's offspring. Or potentially that elicit certain sort of parental behaviors that advantage success. And then finally, there's the possibility that these are spurious associations, and essentially what we're finding is a residual genetic signature of having well-off ancestors. And that third possibility is something that genome-wide association studies are explicitly designed to guard against, but they may have done so imperfectly. Okay, so here's the data. We were interested in understanding how we might distinguish among these explanations, which are not necessarily mutually exclusive. So we assembled life course longitudinal data from 5 studies in the United States, United Kingdom, and New Zealand, tracking about 20,000 people over about 1 million person years, with the aim of asking the question, do genetic differences between people, do carrying more of these education-associated alleles predict moving up the social ladder relative to where you were born? And further, because many of these studies include siblings, we can ask that having more of these education-associated alleles than your brother or sister predict achieving more socioeconomic success than they do. Those analyses address that concern about the spuriousness of genetic associations. If you can compare 2 siblings, the observation that the one who carries more of these genetics ends up better off than the one who carries fewer of them will rule out the conclusion that these are purely spurious associations. So what I'll show you first are some descriptive data from 4 of these studies. The National Longitudinal Study of Adolescent to Adult Health, something local here, the Wisconsin Longitudinal Study, the Health and Retirement Study, which is a national study of adults in the United States, and the Dunedin Longitudinal Study in New Zealand. And what I'm showing you here in these plots are 3 things. They all show roughly the same relationship. The y-axis is the socioeconomic attainment of the individual. Although the dots that are plotted here are not individuals, they're groups of about 50 people in most of the plots, and then the New Zealand plot, there are only 10 of them in each dot. The socioeconomic attainment is measured as occupational prestige, so a rating of the average income and education for a particular occupation then assigned to the person who holds that occupation as an adult. And these plots are stratified by the socioeconomic circumstances into which these children were born, into which they grew up. And those exposures are defined by information about parents' occupation, their education, their income, in some cases welfare benefit receipts, in other cases reports of economic problems the families had during childhood. And what we find is that in the first case, those socioeconomic origins are predictive of people's socioeconomic attainment, something that is widely known. You can see that the cluster of dots under the high SES heading tend to sit higher on the y-axis than the cluster of dots under the low SES heading. The second thing I'll orient you to are the box plots at the very bottom of each of those scatter plots. And those are showing you distributions of this polygenic score within each of these social strata. And it's subtle, but what you find is that these genetic distributions are shifted depending on the conditions into which you're born. Rich kids tend to have more of these education-associated alleles, poor kids tend to have fewer of them. But importantly, what the red regression lines and the scatter plots are showing you is that regardless of the conditions into which you're born, carrying more of these education-associated alleles is predictive of moving up the social ladder relative to where you were born in life. And these are effect sizes for outcomes including educational attainment, occupational attainment, so that prestige measure, and in the case of 2 of our studies, household wealth. And you can see essentially the same result in all the cases. The dark blue dots are showing you the simple association between genotype and outcome. The light blue dots are showing you the mobility effect size. And you can see that the mobility effect size is slightly smaller than the raw attainment effect size. And we see essentially the same pattern of results when we look at comparisons of siblings in the same families. Associations observed within families are smaller in magnitude than our associations observed between families. I'll also orient you to the x-axis in this figure, which is showing you the effect size in terms of a Pearson R. And so the point here is just that all of these effects are small. We're explaining at most a few % of the variance in these outcomes with the sum total of our genomic knowledge to date. Okay, so what does that tell us about our 3 explanations? I think what it tells us in the first case is that we can rule out spurious associations as a pure explanation of the findings in the educational attainment GWAS. Differences between siblings and their genetics predict differences between siblings and their outcomes. Those siblings, by definition, share identical ancestries, and therefore that sort of confounding is not a possibility. They provide evidence instead for these direct effects. In which genetics shape traits and behaviors that influence mobility and success. We also see a suggestion of the possibility for indirect effects, in which genetic associations being partly explained by family context may suggest genetic effects on family processes that advantage children or not. I'm going to skip through an analysis here, except to highlight that if we have data on mothers and their children, what we find is, independent of the child's genetic background, the mother's genetic background is predictive of that child's attainment. There's some caveats to the interpretation of this association that we can get into. I imagine Jason has some thoughts on it as well. But what I will say is that they are consistent with a hypothesis that there are effects of a mother's genetics on her child's outcome that are independent of direct genetic transmission, that instead are mediated by behaviors or environments that are associated with the mother's genotype. And those would be prime targets for interventions aiming to modify children's outcomes. Okay. Very quickly through the takeaways, there are real genetic differences between people that influence social attainments. There is no individual gene or genetic variant for social success. There are lots of them, and each have tiny effects. GWAS have identified some of these, and they allow us to explain a very small amount of variation in the outcome, provided the person has European ancestry. We, as of yet, have no information on genetics for educational attainment influencing other ancestries. Predictive accuracy is poor. Anybody telling you that precision education is around the corner on the basis of these genetic discoveries is overselling it. So that said, even though, as of today, genetics cannot give us precise predictions about individuals, they can help us understand populations. They provide us potentially new tools to help answer old questions like why some people get ahead in life. And that knowledge ultimately has the potential to inform programs and policies to promote upward mobility. Thank you.
Thanks. So I'm back. I'm Jason Fletcher. These are my credentials here. I want to thank Senator Cole, who had a gift to La Follette to do outreach of this type, and this is one of the several talks I'm doing related to an outreach grant for this purpose. So I thank that gift. So I want to start with what I've been thinking about and then shift to policy pretty quickly. I wrote a book with a sociologist, Dalton Conley, that we finished up about a year and a half ago. I just put this up as a reminder to me of what the next steps were. So this is now almost a year and a half old, which now seems like ancient history in our thinking about it. So we have a chapter on the future, and we failed. But when we wrote the book, we thought that we were, in some sense, in the middle of the social genomics revolution. Now, I think you can tell from what Dan was talking about and what I'll speak on to today that I think we're kind of feel like we're back at the beginning. But it does suggest to me, in thinking about it and having conversations about these topics, that although we're not in the middle or at the end of a social genomics revolution, we're in the beginning, we're long overdue in the policy discussions around what Dan was discussing and what I'm going to discuss today. So I think we're already maybe 5 or 10 years behind of thinking about how we want to structure policy around the use of the information that Dan outlined and what I'm going to extend. So I have a brief, so the claim that there's a social genomics revolution, I think, needs a genomics revolution as a starting point, and I have some key items that I think create a social genomics revolution. So the genetic facts that Dan described about differences among people and how genetics might play a role in many social outcomes of interest. But the ones I'm going to just highlight for a slide or two are how the revolutionary part. So the genetic architecture of humans has been the same for a long time. What has changed? So I put them in two buckets. Dan already outlined the second one a little bit or quite a bit about statistical improvements. Let me just bring us up to speed about the technological improvements that have been so vast. So the two key buckets here are unprecedented. I keep using words that that are meant to have a reaction to say, no, this is not unprecedented. There are precedents for this, so let me describe that in a minute, about the dramatic reductions in costs of collecting and measuring human genetics. That this is something I think that is not so well known outside of research. And then a consequence of that are the large and growing databases that are used for data mining purposes and predictive analytics in the way that Dan was describing to create polygenic scores. So let me describe what unprecedented means. So the cost per genome in the last of sequencing the human genome in the last 15 years have fallen by a factor of 100 million times. So that's the unprecedented part. So if you have a counterexample, I'd like to hear that. The closest one is Moore's law, which is the capacity of computing to roughly double every 18 months to 24 months. And you can see that it's not even 15 years. So I described 100 million time reduction in cost in the last 15 years, but you can also see that a lot of that reduction has been in a shorter period of time, maybe the last 10 years. So with that type of reduction in cost, there are many implications. One of them is just the amassing of human data, of human sequence. And so part of that enterprise is from private companies. So this is 3 big players in the United States, 23andMe, with some basic information of how many people they may or may not have collected information for, and the growth. So I've got a couple periods of time, short period of time for 23andMe to go above 2 million. Ancestry DNA claims that they had maybe 4 million people a year ago, and over a weekend last year for Thanksgiving, they had another 1.5 million people because they had a sale. And then a third, runner-up is family tree DNA of over 1 million people. So just those 3 data sets, you're getting a lot of people in the United States, maybe 1 in 30, in 3 private data sets. But what's also interesting is just the future projections. The costs continue to fall in sequencing human DNA. It's very cheap now, and so just the projection, the red line here, which is just the business as usual, not even taking into account the more recent reductions in costs, are 2 billion human sequences by 2025. That's a lot, and it's probably you might imagine it would be higher than that because of how much cheaper sequencing has gotten just since the last few years. So there's the private industry, there's the industry side of it, but there's also tremendous government efforts to amass data sets of humans. Here's a table of these type of programs where so both in the United States and abroad, a couple relatively recent initiatives in Finland, so 500,000 people. New South Wales, they're wanting to get 3 million people. The U.S. has all of us. 1 million people is the target, which you could think of as a weekend's worth of ancestry DNA data collection is our 10-year target for all of us. But what's going to happen with all this data, or what has been happening, and this is getting back to what Dan outlined, which is the use of combining these data sets into very, very large, big data sets to then run genome-wide association studies, which Dan outlined, where it's a data mining exercise to examine across the genome where there might be instances where having one nucleotide versus the other might confer an advantage or disadvantage for health or socioeconomic outcomes. And Dan stole 2 of my figures, but I do have one that's related, which is the time path of GWAS plotted a different way, which is there's been over 3,000 of these. So if you're not in this literature, you might know that there's this groundswell that every day, so this is 1 per day. There's 1 per day GWAS coming out, and there's the typical case would be tens or hundreds of thousands of individuals on a new or old phenotype. So this is just a restatement of some of Dan's pictures of 40,000 or more areas in the genome that have been linked to something important, either on the health side or the social science side. And then to recall what one might do with tens of thousands, 100,000, or 1 million hits across the genome. Well, one thing to do is to summarize them into a single score, polygenic score, and then use this. So the cartoon on the lower right is a potential use in the clinic where you have a patient and they've already had genotyping done, and in combination with the office visit, you might have a polygenic score in front of you for their cardiovascular disease risk. And you might use that, the clinician might use that information to think about urgency of care or even what drug to use. But now I'm going to start shading into policy, and one way of doing that is to just note that these scores are not trapped in the clinic. This is not the only time in your life that you will have some use of these scores. In fact, when you give your saliva sample to 23andMe, they'll send you right back your DNA. And you can do a lot of things with that DNA. One is you can upload it to a number of websites. This one is reputable and non-reputable. This was a good one. This is a group of statistical geneticists at Columbia and NYU. And you'll upload your DNA, and they'll spit back polygenic score of whatever you want. So here's a polygenic score of this particular sample, which 23andMe won't provide, but other places will, like this website. And then, so this is now moved to personalized, out of the clinic, polygenic scores. And then I think there's a set of policy questions of what we want actors to do, both you with your own scores and every institution and organization that you might come into contact with and what they might do with your scores. So that's what I want to spend a little bit of time on. So there's the obvious route of Facebook sending you a lot of products or companies who want to tell you how to live a better life. So this is what type of exercise might be best for you based on your DNA, based on a polygenic score. If you see the fine print, it'll even tell you what wine you might like. So there's also, there's a possibility for regulation of just junk science, as there always is, of just false claims. But there's also maybe less, there's a route that's not just junk. There's a large health component, a precision medicine component of, again, outside the clinic, being able to pay this company is $200 to get your breast cancer risk. And then, so with this proliferation of companies and the way we interact with different institutions in our daily lives, I think there's a set of questions to then ask: how do we want to constrain these institutions versus not constrain these institutions and using this information? So the bucket I'm using is, this is going to allow a new, well, I don't want to overuse new, a direction of discrimination on a different scale, maybe, where a strong, I guess I'll claim, a strong movement of a group-based discrimination of race, ethnicity, age, sex, sexual orientation, and so on, to a personalized. So it's not you because you're a male or a female or whatever, it's you because your genetic score looked like the following. Okay, so then what kind of policies do we want to promote in deciding which institutions are allowed to use these scores and for what purposes, including yourself? So a couple examples that might be obvious to you. Do we want health insurers and employers to use information? So these are two of the actors in the US that cannot use this information because of a law called GINA from 2008. I put this headline as the slide because a year ago GINA was almost destroyed. So would have allowed then health insurers and employers to, yes, go ahead and use genetic information of your employees and your potential insurers. So how do we want to strengthen those type of regulations, broaden them? Life insurers are not under this law. Do we want to broaden it to life insurers and so on? A little bit broader, dating and marriage components of our lives. Nothing right now would stop OkCupid or some other web portal that's meant to have, whose business is to form dating matches, to use your polygenic scores. Your polygenic scores for Alzheimer's disease, education, IQ, you name it. So that's another area of what do we want people to use this for, and what don't we want people to use this for. And then a little bit broader and maybe a little bit more consequential is what are humans likely to want to predict based on these scores? And I'll just remind you all that the scores are based on nucleotides in your DNA that you've had your whole life and you'll always have forevermore, and all the way back in time for when you were a fetus or an embryo. So what might you want to know using this scoring procedure about a fetus or about your baby? So this is another direction that is essentially unregulated. So do we want to regulate anything at all about the knowledge one takes from a genetic scoring based on sex, eye color? There's some easy scoring of whether you're going to have blue eyes or not, or you're going to have a certain color hair, or height's relatively easy to predict, weight. But then as the scoring gets better, so I brought up the amassing of data. Part of that is to produce better scores for a larger variety of outcomes. So that's the direction that we're heading. So which directions do we want to cut off, legislatively or otherwise? And would parents want to know any of these or all of these features of their future children? Is there any evidence from history that parents have ever wanted to know this? So this is a nice cartoon that Dr. Seuss patented exactly this. It wasn't using DNA, but it was for parents to figure out what their kids are going to look like. And the technology was different, which is to take pictures and overlap them and give them to the future parents, and this is what your child is going to look like. So the parents weren't very happy, that their daughters were shown to have mustaches and so on. So this failed. This was a patent that ended up not selling well. But then you ask, well, is there a deep desire for parents to know this, though? And is it only technology that's holding us back? So here's the technology. Here's facial features using only DNA. Do we want parents? So this could be done from a genotyping of a 5-year-old, a 30-year-old, or an embryo. Will parents want this? What will parents want? What won't parents want? What should policy dictate that parents are able to collect and predict? So in that general vein, thinking about IVF procedures is another direction that is essentially unregulated in this regard, where very early on in the formation of the embryo, you could take a genetic sample, and right now this is used to screen out genetic diseases. But the same technology can be used to do anything that I've described, and any kind of polygenic score you can imagine can be done at this stage. So there's no law about what can be used to select embryos in this stage. So it could be what your kids are going to look like, or it could be anything else. And we're in the policy discussion, just to acknowledge that U.S. policy need not be other countries' policy. We haven't talked about other countries yet, except for New Zealand, I guess, but not in a policy realm. But there's I guess there's a general thought in the community that other countries have very different histories. We were talking about the history of eugenics. Other countries just don't have so many, they just don't have the same history. Germany has a very different history, but maybe more similar to the United States, but China does not have that history. And I mean, the projections are, I guess my last point here, that the use of this pre-implantation genetic diagnosis is rapidly growing in China, much more so the United States. So some general thoughts on the future, and then digging into this a little bit more, is the direction is increases in the capacity to predict, much larger data sets, more fancier statistical models that are going to be done in service of prediction. So there's large increases and at the same time, who knows what the actual number will be, the projections of 2 billion human genome sequences collected by 2025. It could be that large, it could be larger, it could be smaller, but the direction is large data sets. So it allows many discrimination, quote unquote, opportunities of the use of this data, and I think it's up to policymakers and the citizenry to think about, well, in what ways do we want this information used? And this is re-raising what Dan described in his talk about the difference between maybe some of the optimistic view of precision medicine using human genetic difference to facilitate targeting of health regimens and interventions versus precision policy, which is like the same genotyping, and then are we going to stop at the hospital door, at the clinic, and say, yes, you can use genetic data in the walls of the hospital, in the walls of the clinics, but not outside? In particular, in which of these or more areas might we want to say yes or no, or leave it up to the companies themselves maybe. So we've had some no's for employers, health insurers. Right now there's not a no for life insurers. There's not a no for schools. And then even for parents, there's not, future parents can presumably get this information if they're savvy enough. What about family members? When I get my genetics, that does reveal information about my family. What kind of firewalls or not do we want for that type of information? So this is all a lot of what do we want? And this is going back to my first point of I have not heard these conversations, and I think that they're about 5 years or 10 years old. This is something that we should have in some sense thought about many years ago. And then something I haven't touched on but that might be of interest in Q&A is in terms of access to technologies. And this will vary across countries. The United States has a unique healthcare system versus many other countries. And what's provided or what's provided through insurance and what's not is a matter of policy. And so when I was describing the use of IVF and the use of IVF related to genetics, is that going to be covered? On one hand, you might be able to screen out a lot of genetic conditions that are very costly from the health insurer's side. That might be something the health insurers would want to do. But for those people who don't have health insurance or who don't have good health insurance, is this just off the table for them? And then I think I welcome this in Q&A is in what instances do we actually want to target policies and interventions around this type of information, these polygenic scores or otherwise. And I'll raise some recent news items that might get us thinking again of what's coming. One is the privacy aspect, which is, you all heard many months ago about the millions and millions of people over a data breach that had their very important information, social security numbers, driver's license addresses, hacked into and released to the public. How far away until this is just what happens to the 23andMe database or any of the federal databases? And there's a difference between your social security number and your former addresses being hacked. Social security number is important, it's permanent for you, but it's not predictive of your outcomes. DNA is permanent and predictive of your outcomes. So when that gets hacked, when, not if, what kind of, again, guardrails are we going to have for the use of that information against the people who've had their information hacked? And this is, in some sense, the variation on these same themes about access to technologies and winding it a little bit, in both thinking about potential increases in inequalities in health, depending on who has access to some of these technologies to the extent that they're beneficial. Another element is the possibility that as we screen more and more, especially for some groups and not others, will we have a general trend towards genetic diseases instead of being common across all groups of people in the United States, being focused only for people who didn't have health insurance or for people with low resources, or another group or certain religious denominations have very strong opinions of the use of these technologies. So this is another policy and social question. I'll just highlight the PGS the procedure I was describing earlier is, from what I understand, completely unregulated. Is that what we would like for the status quo to be? And then I'll say I'm very happy to say that UW is really thinking about this particular set of questions. This is one set of evidence that we'll be hiring at least 3 people in the fall to think about these questions. So we have an initiative on social genomics that several people in the crowd are going to be focused on. And this is exactly the type of questions that we'll be thinking about, and also how new discoveries will translate into the research questions that we are interested in, both from the policy side and from the basic science side. So I hope that this is giving some thoughts on questions. I'm going to wrap up by thanking my funders and local centers and institutes, and I think we have some time for questions. Thank you.
You want to get
Thanks, Jason. And what's your first name? Belsky? Dave?
Dan.
James. Dan. I wasn't too worried when Dan was talking about predicting educational outcomes because I could see the two tests of social science, yeah, significant, but the effects are small, so I wasn't too worried. But when you get to health outcomes, it's pretty clear that you've got, just like artificial intelligence, you've got a real two-edged sword here. It could be used in a positive way to make people much better off, and it could be used in a negative way to discriminate against people, and for various reasons. How? Maybe Alta knows the answer. Maybe she can tell us next panel or something. How do you go about setting these guidelines? Where do you start? And can you, just like with the artificial intelligence use, it's a tool and it's out there and it can be used for positive or negative effects. And how do we think as a society about what's good and what's bad and what is and isn't? I'd be interested in maybe Dan, I'm sorry for butchering your name, Dan's consideration.
It's a hard name. Maybe I'll offer a sort of a very limited response, and then Jason can speak about the substantive policy question. I think you said the effect sizes are small for educational attainment, but health is maybe a bigger issue. And I just wanted to point out that, in fact, the effect sizes for educational attainment, the degree to which we can predict educational differences between people, aren't that different from the degree to which we can predict health differences between people. So genetic tests for Alzheimer's disease, with the exception of the ApoE4 polymorphism, are actually substantially worse than this genetic test to predict educational attainment. They're presently limited to people of European descent, which is a huge limitation. But then I also wanted to make the point that we already have lots of information that is highly predictive of these outcomes for people. It's fairly easy to observe. We can either measure states of individuals at a fixed point in time, or we can measure characteristics of their parents and relatives. And those things already give us this kind of information and in theory are available. Now, I think Jason made some excellent points about how DNA is a very different quantity, and I think it's powerfully true, especially with respect to embryos, but I'll let you take it from there.
I don't have a lot to add except you're right. I think you're right that these are open questions. I've been thinking about this for a little while, and each time after a talk like this, I realized how little advice I have to provide. I think because of, I look to people like Alta and people with other training to think about those next steps. I'll bring out some of the pros and cons maybe, and then think, is the mechanism through votes? Is there other regulation that we might want to consider? But I agree with your general point about the difference between health and socioeconomic outcomes from a prediction standpoint are different. But yeah, I'll stop there. I agree with that point.
Can you help us non-experts understand how to think about genetics given what we're learning about epigenetics? When we're talking about complex, multi-determined traits, we tend to think of genetics as being that stable thing that's part of us from birth. And how do we think about this idea that genes only matter if they're turned on, that genes can be inert, genes could be partially active or very active, which is in itself an interaction with the kinds of experiences people are having very broadly?
So I think that that comment about the epigenome, all of the genome biology that occurs in between a DNA sequence and, let's say, a simple intermediate phenotype like gene expression, is one of the reasons that making clean biological sense of DNA sequence variation in isolation is extremely difficult. It's why thinking about genotypes and trying to understand them may not be a sufficient approach to how we come to understand the genome. I think when we think about polygenic prediction, the epigenome potentially is an interesting mediator, but I'm not sure it substantially changes the way we think about genetic influences on phenotypes, because what we're observing are these genetic effects across a wide distribution of potential epigenetic states. I'd also observe that the a principal determinant of epigenetic variation is the DNA sequence itself. So a lot of what the DNA is encoding is how the epigenome ought to behave. So I think that it is important, obviously, but there are many other exposures outside the genome entirely that affect whether your DNA sequence determines a particular outcome. And so, the classic example perhaps being genetic risk for myopia and vision. Wearing glasses is a perfectly sufficient amelioration of that risk. Genetic liability to alcoholism in a country that prohibits the sale of alcohol is another instance in which a non-genomic factor is intervening between a DNA sequence influence and a phenotype. So I think you can sort of pile the methylome in with all of those, at least to the extent that we're talking about this kind of aggregate genomic prediction, which is to say DNA by no means is the destiny for any phenotype, or almost any phenotype. Complex phenotype.
Thanks. So thinking about this from a very biological perspective and the educational, the polygenic risk score for education, has there been any kind of gene ontology or functional analysis to see what the cluster of genes are that are associated with education? So what are these genes and what do they mean and what do they, and maybe we don't know what their structure and function is, but to me that would make a little more sense than sort of our genes are related to our education.
So there are two answers to your question. One of them is, it makes perfect sense. If we look at annotations of these SNPs to the protein coding sequences that lie near them, and then we annotate those genes to the tissues in which they're expressed, we can find a surplus of genes expressed in the brain and in neural development. So that's a very tidy story. I think there are also many other stories you can tell with these data. The most compelling of these being a paper that Jonathan Pritchard and his colleagues recently published in which they advanced what they called an omnigenetic model. And the observation there was essentially that you can look for tissues in which genes are differentially expressed, but typically what you find to be the largest category to which these SNPs are annotated is generally expressed. And so it turns out that, being well educated depends on lots of things going right for you. Things going right in the brain is good, but things going right in lots of other tissues in your body are also critical. And for things to go right in the brain, things have to go right in lots of other tissues in your body too. And so it's for that reason that genetic influences on an exceedingly complex phenotype like education, or I would argue an equally complex phenotype like having a heart attack, are likely to be distributed across human biology in ways that make it extremely difficult for us to isolate large effects, specific genetic causes that have clean biological meaning. There are going to be exceptions. Those are going to be extremely high-value targets for biomedical science. But as a general rule, when we see a polygenic signal for something like education, I don't think it's going to be something that we can cleanly annotate to a small number of discrete biological processes, or at least that's what the data so far suggest.
Hello. I'm going to drag you guys back to sort of the realm of the first question a little bit, just because I'd love to hear some more thoughts on it. But there's usually talk about policy and regulation at the intersection of industry, application, and sort of the consumer. But I want to know, what can the average researcher do, and how much is it their responsibility to present their research being used for, to prevent it from being used for discriminatory purposes? And is there policy that can be enacted on the research side?
I think it's incumbent on all of us to talk about the limitations of the work that we're doing, to acknowledge the risks inherent in this kind of stuff. I think Jason and I sort of lean in slightly different directions on why it would be bad if people used this information, or how it could be bad if people use this information. Let's say how it could be bad. I tend to think that most of the information isn't very good for individual prediction and isn't likely to become all that good for individual prediction across a broad range of individuals, regardless of how many folks we throw into our genome-wide association studies. Some evidence to that point comes from the relatively small increment in predictive precision achieved in moving from genome-wide association studies of education in 300,000 people to genome-wide association studies of education in 1 million people. So again, that's the yet-to-be-published result, but there's a discussion in that forthcoming article that as we increase the size of our samples, we increase the heterogeneity of those samples, both in the environments that they're exposed to, in the architectures of the genomes that are included, and that limits the gains in predictive power within the genome-wide association study and subsequently the precision of prediction in the polygenic score. So I think that sales of snake oil are a huge risk, and discrimination on arbitrary characteristics, which we have plenty of already. So maybe that doesn't matter in some sense. Like as long as people believe it, that's the problem.
I guess so. There's, I think, I hear the scores are bad enough that people won't use them.
No, I don't. No, that may not be true.
Well, that's as a solution. Like step back. No one's going to use them because they're not very good. Versus cut off the use of them now in the areas we don't want them used.
Yeah, I don't think that's an argument against regulation, or I wouldn't advance that as an argument against regulation.
Because there's going to be instances, this will be our our final comment, thank you, Bobby. There'll be instances where the desires might be very strong to use them. If you think about children before they enter school having potential genetic risks for a series of language development difficulties before they even speak, and whether there's early interventions that might be useful. There's a desire for that, if not the scores necessary to accommodate that. So that's, I think, a question of what we want to allow the use of and what we don't. So thank you very much for your questions. We're on to the next session.
All right, we're going to move quickly on to the next session. I'd like to introduce Alta Charo, who we are really lucky to have here today. She is the Warren B. Knowles Professor of Law here at UW-Madison. She's authored or contributed to over 100 different articles, and what's fascinating part is the breadth of what she covers: environmental protection, reproductive health, new reproductive technologies, medical genetics, stem cell research, science funding, and research ethics. She has served in some pretty big roles. She was a member of President Clinton's National Bioethics Advisory Commission, President Obama's HHS transition team, as well as, she also, you've done so much. It's hard to get it all out. She's worked at USAID, at the FDA, and certainly amongst academics, this is a pretty big deal that she is an elected member of the National Academies of Medicine. National Academy of Medicine. So she'll be talking to us shortly, building on kind of the broader ethical issues with the growing integration of biological data into social science research, and especially social science research that is policy relevant. So I'm going to start off with the sort of final kind of area of biological data that started to be integrated into social science and policy research, which is the gut microbiome. I am actually a professor in public affairs and sociology, and I'm the principal investigator on the Wisconsin longitudinal study, which in part has integrated a lot of these data, genetic data, and more recently data on the gut microbiome. I'm going to be a bit shorter here than my colleagues, in part because the integration of data on the gut microbiome is really the new player in the field, so to speak. This is a pretty new type of collaboration. My collaborator is Federico Ray, who is in bacteriology. He has a lab in bacteriology. And for some of the data and analysis I'm going to present, we also have collaborated with Xingxing Tang, who's in biostats here, whose specialty is analysis of data on the microbiome. But this is sort of the new guy in the field, and in fact, the pilot project that I'm going to talk about is one of the first to attempt to try to integrate research in social science, particularly policy relevant kinds of
policy-relevant kinds of research, basically, where you're trying to understand the role of social contextual factors in terms of how they shape the gut microbiome. But let me sort of start at the beginning. I should say this work is supported by the National Institutes for Health, National Institute on Aging, as well as UW-Madison's Vice Chancellor for Research and Graduate Education Office. Okay, so what's the microbiome? So the microbiome is sometimes referred to as our other genome. It's really important to understand, though, that in terms of the basic science research even, this work is a bit behind the work on the human genome. It's really new, and that's in part, I think, the part of the explanation for why this is still somewhat new for social science to try to think about the role of social contextual factors in shaping the gut microbiome in particular. But essentially, we're an amalgamation of cells, both human and microbial, with a number of microbial cells largely exceeding the number of Homo sapiens cells. Now, there's a lot of recent debate about precisely what the ratio is and so on and so forth, but just note, there are a lot of microbes living on you. In fact, trillions and trillions of them. The microbiome is really different. Well, let me move on and just emphasize, in particular, something that makes the microbiome really distinct. So, in essence, it reflects a shifting paradigm in terms of how we understand microbes and germs in terms of their influence on health, right? Like if you think back to the early 20th century and germ theory, a lot of huge public health interventions, clean water, sanitation, had a profound impact on human life expectancy. If you look at human life expectancy over the course of the 20th century, a lot of the bulk of it essentially was shaped by this theory, in essence, of germ theory, that these kind of microbes were bad for us. This is oversimplifying it a bit. But in essence, part of what this new body of research is showing is it's way more complicated than that. These things are not necessarily, are neither simply good nor bad, but the kinds of microbes that inhabit you appear to be having a fairly large influence on human health and well-being. And this has had a huge influence on basic research. So that's just a recent summary article from the New England Journal of Medicine, sort of summing up particularly on the human intestinal microbiome in terms of its implication for health and disease. But it's also playing a role in terms of how people are thinking about clinical practice. So this is just another study another paper that just came out in the British Medical Journal that's sort of attempting to educate physicians on the microbiome and the ways in which it might be influencing human health and disease. And there's sort of essentially even been medical interventions, particularly the use of fecal transplants. I'm sure many of you have heard about this. For example, to treat C. diff infections in hospitals, which when they become chronic, and this is particularly problematic for older adults, can kill you and did result in thousands of deaths. But this, intervention of these fecal transplants essentially had a fairly large impact on treating C. diff. That's just one example, but there's a ton of interest in this, both in terms of the basic sciences, but then also in terms of clinical intervention. Okay, so let me back up a bit and sort of ground you. I mean, in essence, microbes live all over your body. You have an oral microbiome, a salivary microbiome. I'm going to focus on the gut microbiome, which is where the largest sort of number of microbes are concentrated in the human body. And it's also the case, at least thus far, where there's been really the most research. So again, just to give you some kind of perspective here, I hang out with a lot of demographers, and so they pay attention to people, the number of people, right? So there are 7.1 billion people on Earth, but there are 100 trillion microbes in your gut. It's just a sense of scale, right? Generally, people think about the gut microbiome as a metabolic organ, And it shapes features of our immune system, processes nutrients from our diets, and there's growing interest, although I would argue that the evidence is still emerging, might be generous, in terms of its influence on things like neurological functioning, what's sometimes referred to as the brain-gut or the gut-brain axis. What's key to understand about the gut microbiome, though, especially if you want to think about how larger social contexts might influence its composition in particular, is that you are essentially born, in terms of your gut at least, not exclusively, but largely germ-free, okay? So your gut is colonized within the first 3 to 5 years of your life, okay? And then it can evolve and change over time as you age, but that early life period is a pretty critical period of development. And there's still a lot of debate about precisely how much it changes over your life course, but we know it's strongly influenced by diet. And that period of development early in the life course, right, that the period of which it's colonizing your gut, theoretically, and there's evidence of this certainly from animal models, is that, it plays an influence, for example, in shaping your immune system. So this is why you hear a lot of debate about the role between the gut microbiome and allergies, right? And why we see kids on farms are less likely to have allergies. That's a sort of another example of work that's sort of emerging from this. Okay, there is indeed a large literature that shows a strong relationship between the gut microbiota and human health. But again, I want to emphasize in all these areas, especially inflammatory-related diseases, and immune functioning kinds of diseases. I want to be clear here, though, in the sense that, again, if you think about this, like if you compare this to work on the human genome, this work is in much earlier stages of development. For example, there's a ton of work on animal models. There's somewhat probably less work in humans. And there's still a lot of debate about kind of causal mechanisms. And so I don't want to be overstating here claims about what they know. Okay, but the big question is, why on earth is a social scientist and policy researcher talking to you about it? Well, this is a paper that just in the last month came out in Nature magazine, which attempted to really parse out. There's actually quite a bit of work on this already, but this was sort of it's getting some attention here, this new piece, which basically demonstrates the kind of substantial role environment plays over host genetics in shaping your human gut microbiota. So this particular paper, for example, showed that the average heritability of the gut microbiome taxa is only 1.9%. So in essence, what's a function of genetics, human genetics, is 1.9%. Okay, so relatively small. Okay, now, if you want to ask in question and answer, my colleague in particular, Federico Rey, actually has projects looking at the role of host genetics on the composition of the gut microbiome. But I think it's fair to say, though this is a little bit controversial, by and large, the environment, we know the environment plays a huge role in shaping your gut microbiome. So of course, as a social scientist, that's immediately intriguing to me. And indeed, a lot of the work on the gut microbiome and on what I like to talk about as sort of microenvironments are the effect of microenvironments on the human microbiome. So specifically, human behavior. So you can't open the Wall Street Journal, the New York Times, the New Yorker without finding one of these articles. So these are just examples of like how what people eat influences the gut microbiome, the relationship with obesity, breastfeeding, all these things. So these sort of behavioral, individual level behavioral characteristics, these sort of microenvironments. Now, this shouldn't be surprising to social scientists, to population health scientists, because there's, of course, a robust and large literature documenting how these precise kinds of individual level behaviors influence morbidity and mortality, okay? So it wouldn't be surprising that the gut microbiome might play a kind of a biological mechanism that helps understand those relationships. But there's also a ton of work, which you saw, especially in the first session, showing the relationship between larger environments. So instead it's sort of these microenvironments like what you're eating or what you're drinking or whether you're exercising, but these sort of broader social environments. So poverty, the kind of house you live in, the kind of neighborhood you live in. We now have an enormous amount of research showing that those play a very large role in shaping morbidity and mortality. And as the first panel showed you, there's interest in terms of understanding the biological mechanisms in terms of the brain. There's also a lot of gene by environment work, or a growing body of work in terms of how environments and social environments might moderate the influence of genetic factors on health and other kinds of behavioral factors. So we also might think that maybe those same social environment influences that are so important for morbidity and mortality the microbiome might play a intermediate biological mechanism sort of in the pathway there, okay? And from a policy perspective, I want to emphasize that work on how broader social environments affect health, I mean, I think you got the sense of that from Katherine's presentation in particular, right? Like, we know this matters from a policy perspective. You're seeing places like the World Health Organization requiring countries actually to think about health in all policies. And what they mean by that are how are these policies that alter social environments in turn influencing health? Because we know the social environments matter. So let's think then a little bit farther upstream about how those social policies matter. Okay, so I'm going to do a mini case here to give you an example about how this sort of thing might play out. Okay, but again, remember, in terms of the science, we're in a much earlier period here, okay? But I'm going to focus specifically on the case of social relationships and health. So this is the odds of decreased mortality associated with social relationships. There's been a lot of media coverage of this actually over the past year or two about how social isolation is essentially deadly. This is especially true for older adults, okay? So this is a meta-analysis that's presented quite a bit. So if you look at the sort of odds of decreased mortality, you see that high social support, high levels of social integration are really protective against mortality. Now let's compare that to some of those individual level behaviors. That's smoking cessation for people with cardiac problems, actually. Slightly less effective. How about drugs for hypertension? Physical activity, being lean versus obese. And so there's actually decades of social science research and population health research showing this. And the policy relevance is getting really interesting. I don't know how many of you saw this, but the UK actually appointed a minister for loneliness. And there actually are, if you look, there's a lot of kind of local areas in the UK that actually are trying to implement policies to try to improve social integration and social support for older adults. This really matters, okay? Now there is some really interesting preliminary evidence in terms of the connection between social relationships and the gut microbiome. So there's some work from animal models, for example, especially primate models. Chris Coe, who's actually on the faculty in psychology, has another paper like this I believe it's in psychosomatic medicine, that has a similar finding where you can see evidence of kind of how social behavior is influencing the gut microbiome. And just to help you understand conceptually, mechanistically, this can be anything from if you interact with people, you're exchanging microbes. So we know that your salivary microbiome can end up basically, microbes from your salivary microbiome can end up in your gut microbiome. So you can imagine in intimate relationships in particular, borrowing someone's toothbrush. I mean, there's a million ways. And we see evidence of this in human studies. So for example, there's these really cool studies in NICUs where they track the development of the gut microbiome in these babies in the NICUs, and they're looking at the physical environment in those rooms, right? And what they find is microbes in the physical environment, like on the countertops or whatever, 2 or 3 days later are ending up in the baby's gut microbiome. So we know that mechanistically this is a possibility. This is where I come in and my own work comes in, and that I started this collaborative project a few years back with Federico to try to understand basically, or at least build some data to try to help us understand a little bit better the influence of social conditions across the life course on the human gut microbiota. So we used a sample that's already been mentioned here, the Wisconsin longitudinal study. Just to give you a brief framework here, they are already older adults. They're currently in their 70s, which is when we took these samples. And we have a collection of samples from married individuals and siblings, and then people who are unrelated to each other that we drew from that study to look at this. And the first thing that we decided to look at was, again, to focus on social relationships. And one of the most important social relationships across the lifespan, and this is true in later life as well, is marriage, in essence. And a lot of our folks have been married to each other, in the sample, have been married to each other for 50 or 60 years. Now, there was some existing work that did find, for example, that a couple of studies found that married couples who were living together did have more similar gut microbiomes. So their gut microbiomes kind of looked a bit more like each other than did people who were just unrelated to each other, kind of living out in the community. So there was a relationship there that you've seen in other studies. A third study found no relationship between, a third study basically did not find that, did not find much similarity between married couples' gut microbiomes and unrelated individuals. What we decided to do was compare the married couples in our sample who've been married to each other for 50 or 60 years to siblings to unrelated individuals. And the gist of what we found is that married couples, and they're all folks living together, indeed did have more similar gut microbiomes than did the unrelated individuals, which a couple of studies had found, but also compared to siblings, which was kind of interesting because siblings do share genetics, obviously, and they also shared those really early life environments when the gut microbiome is being established. But to some extent, what was even more interesting and what was really great about using a project like WLS to do this is we could get at the quality of these marital relationships. So if you look at the social support literature, what you do find is that it's not just that marriage is good for your health, it's that good marriages are good for your health, okay? And what we did actually find was the thing that explained why married couples had so much more similarity than unrelated individuals was it was driven by the closeness of that marital relationship. So if you compared people who said we have very good relationships, they did have very similar or much more similar gut microbiomes than did unrelated individuals. If you looked at the married couples, though, who said that their relationships were so-so, they did not look that different than unrelated individuals. There was both a magnitude and statistical significance. So that was another kind of interesting part of the narrative. And we also found, lastly, so one of the things that people look at, and this is, I'm going to say something that's oversimplified here because I'm trying to move quickly, but the general idea is the more diversity that you have in your gut microbiome, the more richness that you have, these are two slightly different things, the better it is, okay? The more varied your gut microbiome, the better probably it is in terms of a range of different health outcomes. And so the brief example that I'll give you this, the rudimentary example, is Federico talks about this experiment on mice he did when he was in grad school, where they fed some mice like the regular chow, like what mice get, and then they fed the other mice only processed food from they went to like the 7-Eleven and just kept feeding the mice that processed food. The mice that got the processed food essentially died off really quickly. And they certainly did see evidence of a lack, basically lower variability in kinds of the diversity and richness of the gut microbiome in those mice. So that's just a rough example. But the gist of this slide is that we also did see that married couples did tend to have, compared to people who were living alone, did have more diversity and more richness in terms of the composition of their gut microbiome. Okay, so in conclusion here, this is an area where because of WLS, I've put my toe in the sociogenomics literature as well, which is, I'm always like blown away about how things change from one month to the other. I think this is also true for work on the gut microbiome. So the science is rapidly evolving. So a lot of what I've said here is really preliminary. But what I want to emphasize is it's not just potentially about social relationships. There are a bunch of other really intriguing studies finding evidence about how physical environments, for example, like pollution, might influence or is influencing the gut microbiome. That's both from human studies and from animal models. There's evidence about housing conditions and how those might influence the composition of the gut microbiome. So I think that this is an area that might be rich for social scientists interested in social environments that can be modified or altered to start maybe looking a little bit closer within here. So for example, I'm one of those people trying to convince Katherine to add fecal samples from the infants in her study. So there's a lot of room here, I think, for kind of new research to get a little bit of a better understanding about what's going on in these social environments and in terms of how, at a biological level, they're actually influencing these morbidity and mortality outcomes that we see so consistently in the social and population health sciences. So I'm going to stop there and introduce Alta, who's going to help kind of wrap up in a bigger way our thinking about this kind of work, certainly in terms of the ethical implications.
First, thank you very much. This is a real pleasure. I think actually I'm going to be the odd duck here because so far we've been talking in many different ways about the way science should be informing policy and should be used to try to help figure out which policies, social policies in particular, would actually be the most effective to accomplish whatever goals we've set. I'm going to flip that on its head because what I'd like to talk about now is how social policies affect the conduct of science, which is a really kind of different question to be asking. And the reason I'm particularly interested in this is that I think we've actually changed in the approach we now have, at least in our attitude towards science, that has some really potentially devastating consequences for some of the research going on now. I'm going to speak in gross generalities, stereotypes, and ridiculous kinds of overstatements because of the time. But if you look at it that way, you can think about the 19th century as a time when we really celebrated the inventive power of science. And we've got the steam engines and the railroads. And speaking as a law professor, you see in the law something that tracks that because you see lots of doctrines that are all about trying to promote industry even at the cost of personal injury or financial fraud. It was all about promoting the inventive power of science. And you look at the early science fiction literature, and a lot of it is also in that vein. But when we move into the early 20th century, we begin to see a bit of a shift in which we now see more public concern about the destructive power of science. We have the destructive power of bombs dropping from the air and of chemical and biological warfare from World War I. And again, you begin to see a change in the kind of legal regimes which reflect culture, because you begin to see more things that are focused on trying to quell the destructive power. And indeed, we become really focused on it mid-20th century with the dropping of the atomic bombs. This is Oppenheimer talking about how now I am become death, the destroyer of worlds. And by the middle of the century, we now begin to move into the destructive power of science in the environmental area. So we've got the Cuyahoga River that catches fire in Ohio. We've got the Silent Spring and the pesticides. And suddenly we've got a raft of laws that are all about trying to regulate science to tamp down its destructive capacities. Clean Water Act, Clean Air Act, various kinds of international treaties on the use of chemicals in warfare, et cetera. And we see this continuing all the way up through about the 1970s. But as we move to the latter part of the 20th century and into the 21st century, I think we've hit a different issue. We went from celebrating invention to fearing the destructive power. And I think now with biotechnology, what we've actually entered is an era in which what we fear is not the destructive power of science, but the creative power of science. And particularly because we're talking about biology, a creative power that goes right to the heart of our sense of self, as well as, for many people, their sense of nature and of a deity. We can look first, for example, in the 1970s. We start with the kind of quintessential kind of risk management conference that the scientists had at Asilomar at the early stages of recombinant DNA research, recognizing this might actually be something where we create a bug that could be dangerous. So we see this perfect segue between the destructive power and the creative power now coming together at Asilomar with an understanding that we need to maybe self-regulate, try to titrate how quickly we actually put this stuff out into the environment and proceed with the research. But it moves on from there, and we had a little bit of reference to this from Professor Fletcher's work. The 1970s and 80s, we begin to see the advent of artificial insemination and in vitro fertilization, and the kind of fear that we are now creating people to specification. Now, I will have to tell you that this is a fear we're going to see repeated over and over, even though we have never actually seen it realized. People who are people who are infertile and have to use these things do sometimes tend to try to optimize the genes they get from the donors. But we don't see fertile people rushing to optimize genes when they actually have the option to take the perfectly imperfect genes of their partners. And we've seen that over and over again. But we see the public fear about being able to design our babies. And of course, at the early year of IVF, that was particularly ludicrous because the best you could do was to pick a gamete donor, a sperm donor, and an egg donor, and then let the randomness of combination proceed. There was very little design there. But as we began to move out in terms of the power that these things had in the area of reproductive technologies, we began to see these fears continue to grow and almost exaggerate. So in 1997, we have Dolly, the famous sheep that had been cloned from some mammary gland cells. She was named Dolly, by the way, because she was named after Dolly Parton, because it was mammary cells. That's always the one thing people take away from my lectures, is that detail. Very quickly around the United States, there were state legislatures rushing to criminalize this area of research, even though it was something that for the moment was completely undoable for humans. And in primates generally, more complicated biologically, as well as having many pitfalls. And it was really being developed to make genetic engineering more efficient in the agricultural sector. If you could successfully engineer one animal and then could clone it, you could get a herd out of one engineering experiment. And it was really about efficiencies for agriculture. But instead, it generated this kind of hysteria. And it was a hysteria that also tended to reify these really fundamental misunderstandings of biology. You had people that were concerned that we were going to clone Saddam Hussein and have more dictators, as if there's a dictator gene. But they were willing to say, well, but if you're going to clone, you should clone Mother Teresa, because they thought you'd clone selflessness, but in fact what you'd probably clone is a tendency to scoliosis, right? So really fundamental misunderstandings of what's going on biologically. And yet these bills passed. We've got 5 or 6 states now that already have this law on their books, and it's never been taken away. And the Congress came very close on a couple of occasions, and was stopped only because of the interplay between cloning and the possibility of not designer babies, but designer stem cell lines for research and for therapeutic purposes. Nonetheless, when we were on the verge of criminalizing this, I used to go around talking to scientists about going from clones, from stem cells to jail cells, was the title of the talk, because of the threat that they were under because of this kind of reaction. Now, more recently, we've now gone to another stage of manipulation of biological material with genome editing, something that has been the subject of a tremendous amount of excitement in just the few years since this really hit the news in 2015 out of the labs from Jennifer Doudna at Berkeley and Feng Zhang at MIT. We've seen an explosion in this technique that allows for a much faster, much cheaper, much more precise way to make very specific changes in a genetic sequence, to add, delete, or alter part of the sequence, and also, interestingly, because I know Seth had raised this question earlier, to be able to edit not just the genes, but also the epigenomes. And with this comes the possibility that we could achieve far greater degrees of control over the outcome of a particular experiment. We really are finally at the point where we can actually talk about designing something as opposed to simply choosing from among options, which is your point about pre-implantation genetic diagnosis, or at least changing odds by having this sperm donor and that egg donor instead of that egg donor and seeing what combination you get, even if it doesn't just, I love that picture of the faces that are going to go across one another from Dr. Seuss. So genome editing, tremendous excitement, but also now, once again, raising a kind of almost hysterical level of concern that this was now going to become immediately adopted by people who are going to try to now finally truly design their babies. They'll take gametes or embryos, and they're going to fix little things here and there, and the fear was that it's going to bleed into more and more trivial kinds of changes, so that because I prefer that my child have a medium complexion instead of a light complexion, I'll go in and I'll change that. And again, we've got decades of evidence that when people are given an opportunity to make these kinds of choices, they do not, in fact, gravitate towards these frivolous choices by and large. These things have always tended to be isolated as either serious medical diseases or very rarely the so-called savior siblings. But the fear is there, and again, if this is really going to get cheap and easy, maybe it is a fear you have to pay more attention to. And CRISPR-Cas9 also has another kind of interesting aspect to it. Up until now, a lot of the biotechnology research really needed sophisticated laboratory facilities and fairly sophisticated training. And CRISPR-Cas9 turns out to be something that can be done in many more settings with far less equipment by people with far less training. In a sense, it democratizes science. And that has a very profound impact on our ability to govern it. Think about the effect that YouTube has had on entertainment, where it used to be it had to be Hollywood that produced a movie. Suddenly, we've got everybody's a movie maker, and there's a platform so everybody can show their videos. It truly changes the nature of the entertainment industry by decentralizing the power, and it becomes much harder to control it. Those censors from the 1950s that could talk to the studio heads and tell them that if you're doing a Star Trek episode, you have to cover her belly button, which was true. Paramount had to cover the belly button. Doesn't work when you've got 2 million people doing their own YouTube thing. Same thing with CRISPR-Cas9. The more that we're getting into an era of so-called garage biology, the more we have to rethink governance systems that will be more appropriate to a decentralized form of science, where you don't always know exactly who's doing it, and you don't know where it's being done. It's very different. It's the difference between regulating how much pollution a factory puts out in one spot on the river versus trying to regulate how much runoff there is from all the lawns collectively in Madison, Wisconsin, into the lakes. Now, CRISPR-Cas9 has some tremendously attractive applications, which is why we're not seeing a rush to criminalize the technology wholesale the way we did with cloning. For example, you could engineer mosquitoes so that they will no longer be able to transmit devastating illnesses like Zika. And you could treat the illnesses. We're already seeing clinical trials on this. Take tumor cells, remove them from the body, edit them, and reinsert them in a way that would then allow them to become kind of little smart bombs that would go back into the tumors and kill the tumors. And this is already the subject of research. So nobody wants to shut this technology down completely. But they are aware of the fact that there does seem to be a kind of slippery slope in its creative capacity. On the left here is something that is these are not with CRISPR-Cas9, this is traditional forms of genetic engineering, but this is glowfish sushi, in which the fish have been engineered with a fluorescing gene, which is a routine thing you do in labs all the time so that you can track your experiment. You add a little fluorescing gene as a marker. Well, now you can make the fish fluoresce, and then you grind them up and you put them into your rice and you roll them up in your nori, and you have glowfish sushi. Go online on YouTube. You can't sell it because the FDA does regulate the sale of foods that are novel, and this probably would attract their attention, but you can talk about it because the First Amendment gives you many degrees of freedom to talk about things. And I know this because when I was at the FDA, I spent 2 very long afternoons looking at that YouTube video thinking, there's got to be some way we can shut this down, and discovering there wasn't. 2 very long creative legal afternoons trying to figure out what I could do about these people, and the answer was nothing. To the right is pigs with glowing noses, again with the fluorescing genes. But there's the glowing rabbit that was made by an artist in Europe, working with geneticists in Europe, and asked why to do it, because I can. Now we get, however, to stuff that is not just maybe frivolous but not very threatening, to stuff that is actually kind of more intriguing. So George Church at Harvard University has for a long time been talking about using genetic engineering, and now CRISPR-Cas9 in particular, to do something called de-extinction, to bring back species that have gone extinct. We probably can, through material left in fossils, as well as material from existing animals that are the descendants of these extinct species, recreate the sequence that represents that species and bring back extinct species, which in some cases would be a marvelous thing to be able to do because we mourn the loss of so much diversity. But on the other hand, is a terrifying thing to do because they're now out of the environment that they usually existed in with tremendously unpredictable ecological consequences. So what seems like a romantic idea is one that actually has the power to create animals that could become a threat to all of us. Then there's rewilding. So many of us here have enjoyed the pleasures of the farmer's market over the summer where you can get all kinds of heirloom tomato varieties, which we prefer because unlike the tomatoes you buy in the supermarket, they actually have some flavor. And the ones in the supermarket have no flavor because they've been, over the years, through traditional forms of crossbreeding and hybridization, they've been developed to have traits that help them last longer, transport without bruising. But along the way, those trade-offs have also included the loss of some of the traits that gave the heirloom varieties all of their deliciousness. Well, again now with gene editing, how about if we take the long-lasting, non-bruising, easy-to-store and transport tomatoes of our supermarket and reintroduce the traits that make them delicious like the heirloom tomatoes, right? This would actually be possible. Maybe not with tomatoes, which turn out to have a surprisingly complex genome. I sat next to somebody from here at Wisconsin who gave me an earful about that one day because I thought tomatoes were easy, but it turns out they're not. But it also begins to make us have to question what is natural. If we could use genetic engineering to actually recreate the very sequence that exists in nature, does it matter that we got to that sequence through modern techniques as opposed to traditional hybridization techniques if the outcome is exactly the same? We are at a point now where we are really in a position to create a lot of interesting things, some of them helpful for disease management, some of them whimsical for the purpose of art, and some of them here for the ability to bring back a lost, almost romanticized, nostalgic age in which we have these kinds of large animals roaming the earth, and our tomatoes actually were something you wanted to eat. This then really does come to the heart of the question. What does it mean when a scientist uses these techniques to create something? And so the question I have is this: if a scientist is doing this, which is in vitro fertilization of a human egg, is it actually conveying a message about this? That is, whether or not you really need to believe in a deity or have some kind of more spiritual attachment to nature in order to understand the origins of life. This goes right to the heart of many people's belief systems and is incredibly threatening for many people because it suggests that maybe we are all that there is, and that we do have complete control, and it's up to us to decide what to do with it, and that there really are no higher powers. That is a very radical thing to be saying just by virtue of doing your experiment, right? Of course, one of the objections has always been exactly this, that in vitro fertilization and cloning and all these other kinds of things like rewilding are all examples of so-called playing God. You'll see that phrase in newspapers all the time, and it's usually understood to mean that we as humans are acting in a way that exercises control over our environment, over other animals, or over other people. And it's the exercise of control that is the playing of God, because that's the role of God. And that is the common understanding in the United States. But what's really interesting is that there's a whole other way of looking at it. I highlighted here a book called Mendel's Dwarf, which is this fabulous book by Simon Maurer, where the protagonist is a hereditary dwarf who's descended from Gregor Mendel, famous geneticist from the 19th century. And at one point in the book, he has a choice, because he's looking down at a bunch of human embryos that he has made with somebody of average height, and he knows because of the nature of the Mendelian pattern of inheritance, half of them probably have the mutation that would have them become achondroplastic dwarfs like him. And half of them would probably grow up to be of average height. And he looks at them and he says, so is it playing God if I choose Because, of course, he has to decide, do I choose the one who's going to look like me as a way to kind of say, I believe I'm okay, or do I choose the one that'll be average height because I'm trying to make life as easy as possible for my child? And he's sitting there thinking, do I have the right to choose? Isn't that God's role? And then he says, actually, in nature, it's all statistics. It's all 50-50 odds. Maybe playing God is rolling the dice, and exercising control is playing human. It's a complete kind of 180 switch on how you think about the phrase and how you think about the significance of the creative process. So it leaves us then with this question. Is it possible that there are some forms of research that go so close to the heart of creating life that they actually become a modern crime of blasphemy? Now, you might say most scientists have no interest in this. They go in the lab, they're just doing their work. And that is true for 99.9% of the research. And it's also true for most of the people even doing this research. Every once in a while, you find the people who will actually fess up to the fact that they're doing the research in order to prove you don't need God, in order to prove that nature is not superior to us and that there are no limits on humans. And in fact, here in this article about Craig Venter, who has come as close as we've come so far to actually creating from scratch a synthesized human cell. Hasn't gotten there yet, but he got pretty far. We've now seen synthesized chromosomes and sequences. There's an article from The Guardian in London about this, Has Venter Made Us Gods? And it calls it another triumph of the only major scientific program driven from the beginning by explicit atheism, right? Now, given that, and given that we've seen this kind of tendency to want to simply shut down research that's overly threatening, like we saw with cloning, and now that we've got all of this kind of public angst about germline editing and the designing of our babies, the question is there any reason why the society can't simply criminalize this kind of research that Craig Venter was doing? Or the next step where we truly will be synthesizing from off-the-shelf materials a human cell from scratch. It led me to actually begin to think about this because I was really very nervous during the early 2000s when I was working with other advocacy groups on trying to protect cloning from criminalization. The lack of a tool, a legal tool to try to use to protect the legality of that research really bothered me. Now, the First Amendment, most people here already know, is all about free speech, and that includes the right to say and also to publish and to write. And so some people have tried to argue that the doing of the research is a precursor to speech. I have to do the research to talk about it. That should be a basis for saying there's some degree of constitutional protection for this research. But it turns out that the argument kind of falls apart. But a different way of looking at it is something that comes closer to that Guardian article with Craig Venter. Is there any way you could say that the actual doing of the research, not the talking about it, but the doing of it, is sending a political message? The very fact that I'm willing to do the research is a political message. It would fall into this category that we call expressive conduct. So when you burn the draft card or burn the flag, you're not using words, but you're sending a political message, and by goodness, everybody knows what that message is. We know because they get very upset with you. And so that is considered expressive conduct, and it gets protection. Government can't just criminalize it or shut it down or censor it without having to leap over all kinds of levels of justification because we have this preference for allowing for the expression, particularly of things that challenge political and cultural norms. That's exactly what this kind of First Amendment protection is for. So the government has to have some kind of substantial interest like safety, which in many of these experiments I'm talking about really wouldn't be an issue. The sheer experiment to try to create a human cell in itself, not yet a public safety issue. Could you regulate it the way we did after Asilomar to say that you can't release it from the lab, you've got to have protections? Absolutely. Regulation, no problem, because it's focused on a concrete goal like safety. But overt prohibition, like we saw with cloning, is something that would be subject to a much higher set of hurdles for the government. I want to conclude simply by saying that I think this is actually important because we are really now at this point where we are going to be able to create a tremendous number of things, all of which are going to challenge really cherished notions that we have about what's special about humans versus other animals, or what's special about mammals versus non-mammals, or what makes Earth special compared to other planets. In every way, this notion of specialness and the relationship to some other force, however you describe it in terms of nature and religion, is going to be challenged over and over by this very small but very interesting set of research experiments going on. And the fact that people can create things is crucial because it is in the process of creating that you truly understand how things work together so that you can then take them apart and tinker with them. This is Richard Feynman, a very famous physicist from Caltech, who left this particular line on his blackboard the day before he died, which was, "What I cannot create, I do not understand." So this is where I think we might be heading. Where Oppenheimer had started with "now I am become death," I think now it's going to be "now I am become life," the creator of worlds. So I end only with my own kind of personal opinion on this, which has probably come out pretty clearly anyway, but the quote from Jefferson, who said that liberty is the great parent of science and virtue, and a nation will be great in both, always in proportion that it is free. Thank you very much.
So we have time for a few questions.
Hi, I was just thinking, the biome and disease and so forth. I was thinking of Jared Diamond's work, Guns, Germs, and Steel, where he found that the Fertile Crescent may have been the first civilization because they had 4 large mammals, the cow, the pig, the sheep, and the goat. So they became immune to more diseases because they had domesticated 4 large mammals. In other parts of the world, there was only like the llama in the Andes or the horse in Mongolia. There were only like 1 in other places. So it turned out that the Spanish conquistadors came to the New World and spread diseases that raised havoc with the Indian populations of the Aztecs and stuff. So what do you think of in terms of the role of this kind of exposure, to animals being a positive for our health?
So there is a lot of interest in that, actually. So the sort of cleanliness hypothesis. So like, for example, even in our data, we have a lot of folks who grew up on farms, okay? And we have a lot of folks who still live on farms. And you very much can see, if you compare the people in our sample who are currently living on a farm who compared to who are not living on a farm, you very much see the point about robustness and diversity, pretty large differences between those folks who are on farms versus those who are not on farms in terms of their microbial, in terms of the robustness and diversity of their gut microbiome. And there's some other really interesting studies. Someone at UChicago, he's at UChicago, for example, who has this study, I believe this was in JAMA, where they compared kids who were on, I'm gonna get the names wrong here in terms of, but essentially 2 different types of Mennonite populations. One that tended to kind of be a bit more removed from the farming, versus another group that really had a lot more exposure. Like they tended, the kids were in the barns cleaning up the cows. So even within those populations, both on farms, but one group far more exposed than the other groups, you saw both what you would think you would see, which was more diversity and robustness in the microbiomes of the kids who had the more exposure, but then also this link about being allergic to things, the sort of allergen issues. So there is a ton of interest in that. At the moment, we're not doing a ton with it other than, looking. We didn't see, what was interesting was that we did not see any differences for people who grew up on farms. That sort of they didn't maintain that diversity and robustness in later life, basically. Like, so if you compare people who grew up on farms versus people who did not grow up on farms, there's no real differences there. In our samples, you can only see that later in life. But what we don't know are things like, well, but how did that, what you're implying, like even if I can't see any differences later in life from what happened in childhood, I don't know how that influenced the development of their immune system or immune functioning or things like that. But there is a ton of interest in that, and a lot of really interesting research going on.
Yes, I might have heard this wrong, but I heard that 99.9% of scientists don't care about the bioethics. Is that what you meant? Or that's simply not true. This is an accusation in AI as well, that we're building AI in different ways and no one really cares, but I don't agree. Or did you mean they don't think about it 99.9% of the time?
First, I want to apologize. I've got massive ear infections, and I currently have unbelievably impaired hearing, so I have no clue what you said. But if you want to answer, or if that was for me, if you can tell me what he said.
It's for you. So I believe, you can correct me if I'm wrong here, but I believe what he was asking was, I think you said something along the lines of, in essence, 99% of scientists don't care about the bioethical issues.
Sure.
And he's sort of challenging that, whether or not that's really accurate, or whether it's, I believe what he said was, whether it's that they just don't think about it 99% of the time, which I thought that was pretty good, actually.
Is that close enough for you? I apologize. This has been an ongoing saga for the last 3 weeks. We've actually seen, I think, an uptick in the amount of interest that a lot of scientists have had in the implications of their work, particularly in the biological area, but I think also now in the area of robotics and artificial insemination, in artificial intelligence. Part of it is because there have been educational requirements like the NIH training grants. Part of it, I think, is because of the pattern over the last 30 years of actually seeing efforts to criminalize whole areas of science or defund whole areas of science. And part of it is coming from the scientific societies. Some of them are actually trying to encourage this. AAAS has certainly been having a set of programs to try to encourage it. I still think it's a minority. I do think that people go into the careers they go into because that's what they're interested in. And if you're particularly interested in politics, you're probably not going to wind up at the bench. And if you're particularly interested in bench work, that indicates you're probably not that interested in social science. So it's changing a little bit.
Following Asilomar, there was a temporary moratorium on cloning human DNA because of the potential for accidentally cloning oncogenes as well. And recently there was a temporary moratorium on certain influenza virus experiments that were going on this campus. Because of concerns about, whether they'll create the next epidemic. Do you have thoughts as to whether these moratoriums should be existing, or, who should be deciding on when there should be a moratorium and when it should be lifted? And
I heard the word moratorium. That's all I heard.
So the idea is, especially around things like, I believe you pointed out the flu and research. I think even Ebola, the kind of moratoriums that happen in terms of certain kinds of research, and whether you think that should be happening, and even if so, how you think it should happen, like what processes should be in place if it were to happen.
Well, you're right that we've had these periodic calls for a moratorium, keeping in mind moratorium is supposed to be temporary, not permanent, right? And there's a fear that moratorium actually becomes functionally a permanent prohibition. But we did see this again when genome editing was being discussed at the first international summit organized by the academies. And the idea was that at least with regard to reproductive applications, there ought to be a moratorium until we understood it better, and until there had been a proper opportunity for people to understand kind of what degree of governmental control is available, and then have some opportunity to decide how to exercise it. So you ask me personally, I have no problem with the concept of moratoria, but I do find myself very concerned when they are started without any kind of set of endpoints, right, where you don't know what the criteria are for lifting it. And the second thing is, frankly, they're unenforceable. This is all about personal persuasion and persuasion within the scientific community, and that's very challenging, especially cross borders, and especially in an era in which governments are often now actively competing to take the lead in certain areas. And so a better way, from my perspective, than moratoria is to actually have very active regulatory schemes that really say, we're going to watch this and we're going to continue to put into place enforceable measures to monitor and restrict actions until proven safe in certain circumstances, and to go at it in a more formal way.
So we're a little bit over, but I want to invite all of you to join us outside for a reception and for more discussion. And I especially want to thank our panelists for being willing to join us and present, and also you all in the audience for coming and listening to us and joining in. Thank you very much.